Hey everyone, and welcome to Miss Estruch Biology and this entire AQA A-Level Paper 1 video in which we're going through all of the theory that you need to know for AQA Paper 1 for A-Level, or in other words, the entire AS level. And don't forget, you can get my workbook that goes alongside this video for you to fill in as you're watching to create a full set of notes completely for free, linked in the description. If time is of the essence and you are more interested in getting exam-specific key marking points, examiner tips, topic summaries, and essay links, then check out my ready-made A-Level notes, which I'll also link for you in the description. And don't forget, if you are pushed for time, you can watch this at ×2 speed. AQA Biology Topic 1. And we start with 3.1.1, monomers and polymers, with two very simple definitions that are here for you to learn. Monomer, which is the smaller units from which larger ones are made. The way to remember this is mono means one, so you've got these single smaller units. Poly means many, but the literal definition that they'd want for AQA is polymers are molecules made from a large number of monomers joined together. These are the definitions as they appear in the specification, which means that is what they'd want on the mark scheme. So the monomers and polymers that you need to know for AQA A-Level Biology are the monosaccharides, for example glucose, and we'll look at all of the other monosaccharides that you need to know, amino acids and nucleotides. The polymers are starch, cellulose and glycogen, which I've put within this row because those are carbohydrates. Then the polymer protein, made up of amino acid monomers, and then DNA and RNA polymers, which are made up of the monomer nucleotides. Now, there are two key chemical reactions that you'll see come up throughout this topic, because condensation reactions are the ones that join molecules together. Hydrolysis reactions are the ones that split molecules apart. But in terms of an exam style answer, here are your bullet points of exactly what you would need to say to get full marks on a question that asks you to define a condensation reaction. It joins two molecules together, it forms a chemical bond, and it involves the elimination of a water molecule. Hydrolysis is the opposite. It breaks a chemical bond between two molecules, meaning it's going to split them apart, and it involves the use of a water molecule. Now, these are your generic definitions. If you are asked in an exam question to describe how a protein forms or how a disaccharide forms, they would want you to give the definition of a condensation reaction. But in terms of proteins, you'd be saying two amino acids together forming a peptide bond. So basically, if it's an applied example, you have to name the two molecules and name the bond. Same with a hydrolysis reaction. If it was an applied example, you should be naming the chemical bond that's broken and naming the molecule that's been split apart and what are the two molecules that's been split into. More on that later throughout this video when we see these applied examples. So that takes us on to 3.1.2, carbohydrates. And here's just a general overview of the different carbohydrates that you need to know for AQA biology. The monosaccharides are the monomers, and it's these three here: glucose, fructose and galactose. Disaccharides are made up of two monosaccharides, and you need to know about sucrose, maltose and lactose. And then the polysaccharides, these are made up of multiple monosaccharides bonded together, and you need to know about starch, cellulose and glycogen. Looking at the prefixes here, we've got mono, that means one, di means two, poly means many. So bear that in mind for all of these. Monosaccharides are one sugar unit, disaccharides are two, poly means multiple joined together. So let's have a look at each of these in the level of detail that you need to know. Starting with the monosaccharides, we have glucose, fructose and galactose, like we just said. These are the monosaccharides. And a monosaccharide is the generic term for the monomers from which larger carbohydrates are made. And to turn those monosaccharides or monomers into the polymers, which are polysaccharides, or even into disaccharides, you would have a condensation reaction between two monosaccharides, and the name of the bond is a glycosidic bond. So in carbohydrates, the bonds that you need to learn for how you join multiple monosaccharides together is a glycosidic bond. Now you need to know the structure and the chemical formula for glucose. You don't need to know it for fructose and galactose, but you do need to know it for glucose. So chemical formula, as you learnt at GCSE, 6 carbons, 12 hydrogens and 6 oxygen atoms. This is the level of detail that you need to know the diagram to show how those atoms are structurally arranged. So you don't literally have to show where all of the carbons are, all of the hydrogens and all of the oxygens. They just want you to see the ones that are going to be of relevant information when we come to the bonding. But just as a bit of information, inside of this ring, this hexagonal ring, we have got these corners, let's call them. And wherever you've got these corners, that is representing where a carbon would be. So this would be carbon, and we actually call it carbon 1. This is carbon 2, carbon 3, carbon 4, carbon 5, and the 6th carbon branches off the top there. But you wouldn't be asked to do that. You just need to know you've got a hexagon, you've got a branch coming off here. Oxygen is at this position in your hexagon. And then on carbon 1 and carbon 4, it's symmetrical. A hydrogen atom on top, a hydroxyl group on the bottom. So you do need to know that that is called a hydroxyl group. Now this is actually called alpha glucose, and that's because there are 2 isomers of glucose that you learn. And for those of you that don't take chemistry, you might be thinking, what is an isomer? Isomers are when you have the same molecular formula, so C6H12O6, but those atoms are arranged in a different structure. So structurally it's different, but it contains the same atoms. So we've got alpha glucose here on the left that we just talked about. Beta glucose is here on the right, and the only difference between them is carbon 1, we have flipped the hydrogen to be at the bottom, hydroxyl to be at the top now. So for alpha glucose, it's pretty much symmetrical with the exception of the branch there and the oxygen. But in terms of what's on carbon 1 and 4, it's symmetrical, whereas on beta glucose, they've flipped around. And that has relevance to the final shape that polysaccharides have when lots of these bond together. So I'll come back to that when we get to the polysaccharides. But first, it's the disaccharides. We've covered now everything that you need to know for the monosaccharides. Disaccharides are formed by a condensation reaction, or the condensation, of 2 monosaccharides. We have a chemical bond, which is a glycosidic bond that occurs. And these are the disaccharides that you need to know. We said maltose, lactose and sucrose, but here are the word equations. Glucose and glucose is maltose plus water. Glucose plus galactose is lactose plus water. Glucose plus fructose will make sucrose and water. So ways to try and make this a bit easier to remember is, first of all of them contain at least 1 glucose molecule, which is why I put those glucose in bold. Maltose has got 2 glucose, so that's what you need to remember for maltose. Lactose is made up of glucose and galactose. That one's probably an easy one to remember because galactose has lactose within the name. And then sucrose is glucose plus fructose. Other thing I've put in bold here is something else that's the same for all of them. One of the products for all of these is water. And that's because they are joined by a condensation reaction, which we said is the joining together of 2 molecules, forming a chemical bond with the elimination of water. So that is why water is one of the products each time. So those could be a good thing to put on a flashcard just to test yourself until you can remember those word equations. So just to show you these condensation reactions, we can see here we've got 2 monosaccharides. We've shown some of the atoms within them, but the ones of interest are on carbon 1 and carbon 4, the hydroxyl group at the bottom here. And this is where the water is going to be removed from. So I've highlighted in blue the 2 hydrogens and the oxygen to make up the H2O that gets removed. Now it doesn't have to be the OH of carbon 4 and the H of carbon 1. It could be the OH of carbon 1 and the H of carbon 4. I just happen to have highlighted these. But the main thing is it's from the 2 hydroxyl groups that the water is removed. That leaves you 1 oxygen atom behind, and that's where we get this glycosidic bond, the carbon to the oxygen to the carbon. And this is actually called a 1-to-4 glycosidic bond because it's forming between carbon 1 and carbon 4. A hydrolysis reaction, so the opposite of this, it's through the addition of water, a chemical bond is broken, so that'd be breaking this glycosidic bond, and that then splits those two molecules apart back to their original monosaccharides. And we can see here the water has been reinserted as our hydroxyl and hydrogen to make up that other hydroxyl group. So last in the carbohydrates then is the polysaccharides, which are created by condensation reactions between many glucose monomers. And you learn about starch, cellulose and glycogen. Starch is found in plants, as is cellulose. Glycogen is found in animals. And the overall function of these are starch is a store of glucose, cellulose provides structural strength to the cell walls, and glycogen is also a store of glucose. But we can have a look at all of those in more detail. This is your summary table of everything you need to know for the polysaccharides. So take a screenshot or pause, or if you have downloaded the booklets that you can get for free in the link in the description, then pause at this stage and fill in this table in your booklet. So the monomers is one thing that you need to know. Starch has alpha glucose, as does glycogen. Cellulose is beta glucose. Now, as we go through this table, you will notice that everything we say for starch, it's very, very similar to what we say for glycogen. And that's because these two molecules are structurally very similar. And that's because they both perform the same function. It's just starch performs this function of being a glucose store in plants, whereas glycogen, it's a glucose store in animals. So there are a lot of similarities between these two. Cellulose has got a completely different structure because it's got a completely different function. So bear that in mind as we go through. So starch, alpha glucose is the monomer. It's glycosidic bonds, but we talked about a 1-4 glycosidic bond when we saw that very basic animation showing a condensation reaction. But you can actually also get 1-6 glycosidic bonds. And carbon 6 is the one that branches off the top of the hexagon. So if you get a bond between carbon 1 of one monosaccharide and carbon 6 of another, that creates a branching point in the structure. So we get 1-4 and 1-6 glycosidic bonds here. Function, we've talked about store of glucose found in plants, in particular get a lot in the chloroplasts. And then the structure. Although I've talked about amylose and amylopectin here, I haven't actually seen those two terms come up on the new spec yet in a mark scheme. But I'm keeping it in just in case and so that you've got the full understanding. So amylose, this is one of the polymers in starch, which is made up of only 1-4 glycosidic bonds between the alpha glucose. And that results in this long unbranched chain, which then coils up to make a helix. Amylopectin is another of the polymers in starch, and this one has got 1-4 and 1-6 glycosidic bonds. So that means you end up with these straight chains with branches coming off them. Now, the advantage of these two structures, linking to the function of being an energy storage, is if it can coil up into a helix, because it's a coil, that means you can then compact it down to fit a lot of glucose in a small space. The branch structure on the amylopectin, that provides a greater surface area of exposed ends of the chains. So if you did need to hydrolyze the starch back into glucose for respiration, then having more branches and therefore more exposed ends means that hydrolysis of starch back into glucose happens more rapidly, which is needed for glucose for respiration. And I actually paused and typed that in here because I've remembered they've got really picky on you saying that in the mark scheme in the last couple of years. So you would have to say rapid hydrolysis into glucose for respiration to get that mark. So highlight that in this table as you write it out. Now, it's also a really large molecule, which means it's insoluble and therefore it's not going to dissolve and affect the water potential and therefore have any impact on osmosis. I'm going to jump to glycogen next because that's so similar. Alpha glucose, again, 1-4 and 1-6 glycosidic bonds. Store of glucose, but this time it's found in animals, mainly the muscle and liver cells. It's a highly branched molecule. So this actually has a higher proportion of the 1-6 glycosidic bonds compared to starch. And this means we have even more branches and you get an even more rapid hydrolysis of glycogen back into glucose for respiration. The advantage here is glycogen is the store of glucose in animals, and animals move. They might have to run from a predator, and for that you need muscle contraction, which requires ATP from respiration. So animals need to be able to hydrolyze their glucose store more rapidly for respiration because they might need to run or fight to protect themselves. Plants don't have that, so that's why they don't have as many 1-to-6 glycosidic bonds. But in terms of the structure linking to the function, exactly the same as it was for starch. Branch structure increases the surface area for more rapid hydrolysis back to glucose for respiration. Insoluble, so it's not going to affect the water potential. You don't actually get this coiled structure, so I'd go for these two if I was asked to give marking points for that one. Then cellulose, this one's completely different. It's beta glucose. We have 1-to-4 glycosidic bonds, and this time we have the function of it provides structural strength to the plant cell walls. Now for the structure here, you have long straight chains of beta glucose, and that's because the 1-to-4 glycosidic bonds form directly next to each other, which we saw in that animation when we looked at where the bond forms. So you end up with this long straight chain. The chains are held in parallel, so you have multiple long straight chains held in parallel by hydrogen bonds to make a structure known as a fibril. And one hydrogen bond is not strong, but because there are so many hydrogen bonds, collectively, they provide the structural strength to the plant cell wall. It is also insoluble, so it's not going to affect the water potential. One extra point just to add here about the structure that you'll see is every other beta glucose in these long straight chains is inverted 180 degrees C, which basically means it's flipped upside down. So we can see here the CH2OH is at the bottom, then it's at the top, then it's at the bottom, then it's at the top. So that has to happen in order for you to get this continual chain of 1-to-4 glycosidic bonds and these long straight chains. So that's often worth a marking point as well. So how do we test for these biological molecules then? So the test for starch is you add iodine solution. A positive test observation would be that it goes from this orangey brown colour to bluey black. The test for reducing sugars, you add Benedict's reagent and heat. You need to name the reagent and you have to say heat to get this mark. And we can see here, Benedict's reagent is this blue colour. A positive observation would be that that blue colour turns, and there's actually a range of different colours it can turn depending on the concentration of sugar present. So you can go from green to yellow to orange or brick red, and the more red it is, the higher the concentration of sugar present. So this one here is orange, that's quite a high concentration of reducing sugar present. The non-reducing sugars, which is only sucrose. The reducing sugars is all of the monosaccharides, all of the disaccharides except sucrose. You don't need to learn about why that is the case. You just need to know that sucrose is the only sugar that you learn that is non-reducing, which basically means it can't reduce the copper sulfate in Benedict's reagent. So for the non-reducing sugars test, this would be a longer question because there's more stages. First of all, you have to do a reducing sugars test. And if you get a negative test result for that, meaning it remains blue, that tells you you don't have a reducing sugar. So the next step is then to see, well, maybe it is still a sugar, but it's non-reducing. And the way to test for this is by splitting sucrose, or non-reducing sugar, back into its monosaccharides to confirm that it is sucrose. Now to do this, we have to hydrolyze sucrose back into the monosaccharides. And this is done by something called acid hydrolysis. But the marking point would be add acid and boil, or boil in acid. You don't have to name which type of acid. You would then cool the solution and add an alkali to neutralize it, or in fact it goes beyond neutralizing, it makes it slightly alkaline because that is the pH that Benedict's reagent works best in. Then you would have to say add Benedict's reagent and heat. And yes, you do have to say step 4. Sometimes students miss that one out because I think, well, I've already talked about that in step 1, but this is actually now starting again after you've done the acid hydrolysis. And because we cooled it down, you have to heat it up again. So don't miss this mark. Add Benedict's reagent and heat. And this time, the positive test observation, it'll go from that blue to probably orange or brick red. You don't usually get the yellows or greens for this, and this is often an application question. And it's because you should have quite a high concentration of sugar now, because if you started with sucrose, lots of sucrose molecules, which are disaccharides, and then you hydrolyze that into monosaccharides, you've now doubled your sugar concentration, because you might have gone from, let's just make this simple and say 10 sucrose molecules. After we hydrolyzed that now made 20 monosaccharides. So you have a higher sugar concentration. That's why you should get orange or brick red as your positive test result. 3.1.3 is lipids. So the 2 lipids that you need to learn about are triglycerides and phospholipids. Triglycerides are formed by a condensation reaction, and the molecules that you're joining together are 1 molecule of glycerol gets 3 fatty acid molecules attached to it. So you actually have 3 condensation reactions happening here, 1 for each of those fatty acid chains. In a phospholipid, you still have a glycerol molecule, you still have fatty acid chains, but you don't have 3, you have 2, because one of the fatty acid chains is swapped for a phosphate group instead. And we'll have a look at some images of this as well. So the condensation reaction is occurring between the glycerol molecule and the fatty acid for both of these, and the bond that is created is called an ester bond. We talk about R groups with the lipids, and the R group is linked to the fatty acid. And with your fatty acid, the R group can either be saturated or unsaturated, which we'll come back to. But this is just visualizing what we were saying in terms of the structure. So this one here is a triglyceride, tri meaning 3, and we have 3 fatty acid chains. So you have glycerol at the top here. You might be showing it from a different angle, but basically you have a glycerol backbone with 3 fatty acid chains attached to it, which would have been 3 condensation reactions and therefore 3 ester bonds. For the phospholipid, one of those fatty acid chains is not present, and instead we have a phosphate group. So it would have been 2 condensation reactions, 2 ester bonds. And this is just showing you a bit more detail about the condensation reaction between the 1 molecule of glycerol and the 3 fatty acid chains. So here is glycerol. You don't need to learn the chemical structure of it, but we do have glycerol with the hydrogens on this side, fatty acid chains which end in a carboxyl group, C double bond O, OH. And the water is removed from that hydroxyl group and that hydrogen, and we can see that ester bond then is a carbon to the oxygen to the carbon, but with that double bond O and the H and H there. So that forms an ester bond, and they've got that written here, our ester bond. So this is just showing you once more, just to make sure we're all clear on this, because sometimes they do ask you to circle where the water's removed from, where the ester bond would form. So here's glycerol. Here's one of the 3 fatty acids. Here's where we're removing the water from. And that is where the water is going to be removed. There is our ester bond that is made, or the 3 ester bonds. And that means 3 molecules of water have been made because of those 3 condensation reactions. Now we talked about the fact that there are fatty acid chains, and the R group can be either saturated or unsaturated. So R group generally means a variable group, the bit that's different every time. So for all fatty acids, they all start with a carboxyl group, which is the name for this C double bond O, OH. And then you have this hydrocarbon R group, meaning it's just made up of hydrogens and carbons. And it can either be saturated or unsaturated. Saturated is when you only have single bonds between all of the carbons in the chain, and therefore you have the maximum number of hydrogens bonding to it. So it's saturated. But to get the mark, you would have to say only single bonds between those carbon atoms. When you have an unsaturated fatty acid, you have at least one, we've only got one in this picture, but you could have multiple, so at least one double bond between the carbon atoms. And because of that double bond there, that now means we've got bonds used up here, which would have been used to bond a hydrogen. So it is now unsaturated because it's not bonded to the maximum number of hydrogens possible because the bond is used up in that carbon-carbon double bond instead. But for biology, the definition is the parts highlighted in yellow. So you need to know the properties of these different lipids. So triglycerides, they are an energy storage molecule. And the way that the structure links to the function is the energy storage part is the idea that there's a large ratio of these energy storing carbon to hydrogen bonds compared to the number of carbon atoms present. And that, in terms of the chemistry behind this, provides a lot of energy storage. You don't need to know the ins and outs of why, you just need to know that's the fact. There's also a high ratio of hydrogen to oxygen atoms, and they can act as a metabolic water source, basically meaning that triglycerides can release water if they are oxidized. And that's essential for animals in deserts. So for example, camels, their hump is made up of triglycerides, not of water, but they can then break down those triglycerides to get this metabolic water source. Triglycerides do not affect water potentials because they are insoluble in water, therefore has no impact on osmosis. And that's good because we don't want lots of water moving in or out, causing cells to burst or shrivel. Now that is because they're very, very large, but they're also hydrophobic. Now this isn't a term we've come across yet, but hydro means water. Phobic literally means fearing. But in terms of biology, it means to repel. So water repelling, and that's what makes them insoluble in water. Lipids also have a relatively low mass, and that means you can store a lot of these lipids without it increasing the mass as much as, let's say, muscle tissue would, and therefore it's not going to impact mobility as much. So phospholipids, in contrast, these have a very different function. This is just showing us the structure again. We've got the glycerol molecule, two fatty acid chains, and the phosphate group. Now, although this picture is showing one saturated and one unsaturated fatty acid chain, that is not always the case. It's just what this picture is happening to show. So we've got two condensation reactions would have occurred to make two ester bonds. And the key difference here is the properties that this phosphate group provides on what we call the head of this structure. And the head is basically the red circle that we've drawn around the phosphate group and the glycerol molecule. That part is known as the head, and it's described as being hydrophilic. Hydro meaning water. Philic literally means loving, but that's not what we mean in biology. Hydrophilic means that it can interact with water. It's not going to be repelled, instead it can interact. And that is because of this charge on that oxygen in the phosphate group that makes it polar, meaning it's got a charge, and therefore it's going to be able to interact with water, but polar molecules repel fats or lipids. So we've got this hydrophilic head that can interact with water but repel lipids. The tails are known as hydrophobic tails. They don't have any charges on them, so they are non-polar. And hydrophobic tail means it repels water, but it interacts with fats. Now, it's that hydrophilic head and hydrophobic tail that link to the main function, which is the fact that phospholipids form cell membranes. They create this phospholipid bilayer, two layers of phospholipids, and that makes all cell membranes, whether it's cell surface membrane or the membranes on organelles. So in aqueous environments, because of that liquid, that water, the phospholipids will arrange like this. The hydrophilic heads face outwards towards the water because they can interact with it, but they're repelled by the lipids. Whereas the hydrophobic tails are repelled by water on the outside, so they spin inwards and they can interact with the fatty acids on the other side. So that's why it forms this bilayer in this structure. Now, the test for lipids in terms of the biochemical test is you would need to take your sample that you're testing, mix it or shake it with ethanol, then you add distilled water, and a positive test result is white in colour, and we describe this consistency as an emulsion. For this one, it's really important that you don't say add ethanol and water and mix. You have to add the ethanol first, and then you add the distilled water. And in terms of in the mark scheme, they often accept you saying mix and shake at point 1 and at point 2. Whenever I've done this experiment in the lab, I always do it in this order, and you can see you do get that test result there. 3.14 is proteins. So amino acids are the monomers from which proteins are made. And proteins have loads of different functions because there's so many different proteins that can be made. And what's different about all of them is their final 3D shape. And that is what determines the function of a protein. You need to know the general structure of the monomers, the amino acids. So here we have what's known as the central carbon. And coming off that, you have the amine group, NH2, a carboxyl group, COOH. You have an R group, which is this variable group, meaning it's different every time. And because there's 20 different amino acids, there's 20 different options of what that R group could be. And then there's always a hydrogen atom as well. You do need to draw the amine group on the left, the carboxyl group on the right, but it doesn't matter if you have the H or the R on top or on bottom. So the best way that I find to remember this is always draw your central carbon first, which means the carbon in the middle, and then 4 branches coming off it. And then it's just trying to remember amine group, NH2 on the left, carboxyl group, COOH on the right, R and H top and bottom. So as I said, there's 20 different amino acids, and what is different each time is which side chain they have, which is that variable R group. To form a dipeptide, which means di means two amino acids bonded together, it is another condensation reaction. So we can see we've got the hydroxyl group removed from the carboxyl group of one amino acid, and the hydrogen removed from one amine group of the other amino acid. That is where the water is being removed from. The two molecules being joined together are 2 amino acids, and the bond that is formed is a peptide bond. So that is how we make a dipeptide. A polypeptide means many amino acids joined together via many condensation reactions, and many peptide bonds would form. Now, when you do have your polypeptide chain, that is then arranged into 4 levels of structural arrangement. The final finished protein is either tertiary or quaternary in structure. Primary is the first structure that's made, but it has to be processed, folded and packaged. So the first stage is making it a secondary, then a tertiary, sometimes it's a quaternary. So let's go through what each of these terms mean. The primary structure is the order or sequence of the amino acids in a polypeptide chain, and those amino acids are held by peptide bonds. This used to always come up as a 1-mark question. It'd just be that first bullet point. Now, since about 2023, I've seen it come up as 2 marks, and they wanted you to say as your second mark, held by peptide bonds. So let's add that in. And as a key point, they will underline the word order or sequence. You have to say that to get the mark, because the order that the amino acids form is going to determine the 3D shape that we see in the tertiary structure. So that's the first level of arrangement. That then will go to the Golgi body or Golgi apparatus and gets processed further into our secondary structure, which will either be an alpha helix or a beta pleated sheet. That's the name for these 2 shapes. And what happens is that sequence of amino acids that we saw in the primary structure, that goes and gets coiled or folded into these 2 shapes. And the key thing is knowing those are held in those shapes by many hydrogen bonds. So your key marking points of this one would be stating alpha helix and beta pleated sheet held by hydrogen bonds for your 2 marks. And you could be asked, where do the hydrogen bonds form? So it is between different amino acids, and it is between the oxygen and hydrogen. The oxygen in the C double bond O of a carboxyl group, and the hydrogen in an amine group. So that's what I'm showing you here. These pink dashed lines are representing hydrogen bonds forming between different amino acids, between the oxygen of the carboxyl group and the hydrogen of an amine group. Then we get the tertiary structure. So this is often a 3-mark definition. It's the further folding of that secondary structure. So again, this is happening in the Golgi body where you process and package proteins. That forms this unique 3D shape. It doesn't look a lot there because it's just a load of squiggly lines, but that is folding on top of folding to create a 3D shape. And then the 3rd mark, you have to name the different bonds that hold that 3D shape in place. So you have ionic bonds, more hydrogen bonds, and sometimes you get a disulfide bond. Disulfide bonds means 2, di meaning 2, sulfide, so it's the element sulfur. So if you happen to have an R group that contains sulfur and you have 2 of those in your chain, then you can get a disulfide bond as well. But you don't always get them because you only get those disulfide bonds if you do have 2 R groups that have sulfur. Just to point out here, I've noticed I've used different spelling. It used to be disulfide was the British spelling with a PH, disulfide with an F was US spelling. They have now just used the F spelling universally. So PH isn't actually used, F is. You wouldn't get penalised in biology, but that's just why I've got that disparity there. Okay, lastly then, quaternary structure. This is when you have a protein made up of more than one polypeptide chain. And in this example, I've picked 4 because that links to an example that you learn later on in the course for hemoglobin. Each of those polypeptide chains is still folded up into a tertiary structure though. So this is still going to be a unique 3D shape. It's just you've got more than one polypeptide chain doing that. So the importance of the primary structure here, which was our first one, that unique sequence of amino acids held by peptide bonds, is even one amino acid being in a different position or different sequence will mean that the bonds that would form in the tertiary structure, so those ionic, hydrogen disulfide bonds, would be in a different location. And that's because these bonds, the ionic bonds, disulfide bonds, they occur between the R groups. So if you're moving the amino acids in the chain, you're moving where the bonds are going to form between the R groups. And if you have bonds in a different place, that causes a different folding shape, and that's why you end up with a different 3D shape. So this comes up a bit later, more synoptically, when you learn about mutations in topic 4 and topic 8, because the sequence of amino acids in the primary structure is coded for by the sequence of bases in DNA. So if your DNA mutates and you get a different base sequence, you might code for a different amino acid sequence. Therefore, these bonds occur in different locations, you get a different 3D shape. And if that happens to an enzyme, for example, and you get a different 3D shape, that now means you've got an enzyme with a different shaped active site and it's not going to function. And that's what we're just showing here. If it was carrier proteins though, or channel proteins that you have embedded in the cell surface membrane, if they change shape, then the molecule that they transport might no longer be complementary and it can't be transported across the membrane anymore. So that bit I've actually already said, mutations could cause this. So test for proteins then is this bright blue solution biuret and it turns purple. No heat is needed, but the key thing to remember here is the spelling. Everyone that takes chemistry, you are familiar with a biuret that you use in titrations. One common mistake here is writing biuret instead of biuret. They will accept if you've spelt it incorrectly, but phonetically it sounds the same. But I just split it up here just to try and help you remember. Biuret. Don't say biuret, you're going to lose the mark. Okay, enzymes. This is still within the proteins topic. This is one of the key examples that you need to learn about. Enzymes are proteins that have a tertiary structure, and their role is to catalyze reactions, and they do this by lowering the activation energy, meaning the amount of energy needed to start the reaction. And whilst enzymes are pretty big proteins, it's only the active site, that small part here, that is of importance, because that's where the substrate binds. So the active site is specific and unique in shape, And that is because of everything we just talked about there in protein structure. That unique sequence of amino acids, determining the exact location of the tertiary structure bonds, like the ionic and disulfide and hydrogen bonds, causes a particular 3D folding in the tertiary structure to create that unique shape active site. And due to this specific active site, enzymes can only bind to a particular substrate, and therefore they only catalyze one particular reaction, and they will create this enzyme substrate complex. Key marking term, that enzyme substrate complex. And once you get that complex, that is what lowers the activation energy. Now there's two models of enzyme action to explain how the enzymes lower this activation energy. You learn the lock and key model at GCSE. You now learn the induced fit model at A level as well. And this is the accepted model now, the induced fit one. But you need to have an awareness that the lock and key used to be the accepted one. So lock and key, just briefly, this is the idea that the enzyme is like the lock, substrate is like the key, and they fit due to being perfectly complementary in shape. The enzyme never changes shape. So the enzyme active site is a fixed shape. Due to random collisions, we would get the substrate binding, you get an enzyme substrate complex, and once the enzyme substrate complex is formed, the charge groups within the active site are thought to distort the substrate and therefore lower the activation energy. And then the products are released, the enzyme active site is empty, and the enzyme can be reused. It's not used up in the reaction. The induced fit model is the current accepted model. And in this model, the enzyme is more like a glove and the substrate is like your hand. So when you've got an empty glove, it's not exactly complementary in shape to your hand. But when you put your hand inside of it, it enables the glove to mold around your fingers and then it becomes completely complementary in shape to your hand. That is what is happening in this model of how we think enzymes work. The active site is not actually completely complementary in shape to the substrate until it collides and binds. Because when it does that, the active site molds around the substrate and then it does become completely complementary. But because it's molding around it, when it's in this enzyme substrate complex, that is going to be putting strain and tension on the bonds, and therefore less energy is needed to break them. So that is the accepted model. Now we have more of an awareness that proteins aren't completely rigid. There is some flexibility to their structure, and it better explains how the activation energy is lowered. So factors that affect enzymes then are temperature, pH, substrate concentration, enzyme concentration, and also inhibitors. So we're going to go through each of these. Temperature then, if you have too low a temperature, then the enzyme and the substrate don't have enough kinetic energy to be able to move fast enough and have successful collisions, meaning they bind and you get those enzyme substrate complexes. And that's why we can see at the lower temperatures, there's a lower rate of reaction. But as you increase the temperature, the rate increases because more kinetic energy is gained and therefore more successful collisions, more enzyme substrate complexes. So at that point, temperature is the limiting factor, meaning it's limiting the rate, and as you increase the temperature, the rate increases. We then have our optimum temperature, which means the temperature at which the reaction happens the fastest. Beyond the optimum, there is now so much kinetic energy that the tertiary structure bonds will break. So the ionic bonds, hydrogen bonds will start to break. And if you break those bonds in the tertiary structure, the tertiary structure protein is then going to unfold and you lose that unique 3D shape. And therefore the active site's a different shape. The substrate can't bind, you don't get enzyme substrate complexes, and the rate of reaction decreases. With pH, too high, meaning too alkali, or too low, meaning too acidic, a pH, interferes with the charges in the amino acid, and in particular, of relevance is the amino acids in the active site. And this again causes a break in the bonds. So if you've got too high a pH, that means you've got too many OH minus ions. Too low a pH, which is more acidic, you've got too many H plus ions. And because you've got too lots of ions there, whichever it is, Those ions interfere with those charges, causing the bonds to break, the ionic bonds to break there, that are holding the tertiary structure in place. So same idea again, the bonds break, that means the tertiary structure unfolds and you lose that unique 3D shape. We describe that as the enzyme's denatured, fewer enzyme substrate complexes form, and that's why the rate of reaction decreases either side of the optimum pH. Optimum pH meaning the pH at which the rate of reaction is fastest. And the optimum pH is different for different enzymes because some enzymes are going to be working in locations that are very acidic. So for example, the stomach has stomach acid, which is very acidic. So enzymes that work there, such as proteases, are going to have an optimum closer to 1 or 2. But the enzyme amylase has an optimum closer to 8 because the pH of the small intestines is slightly alkaline. Substrate and enzyme concentration then. If there is insufficient substrate, then the reaction will be lower. So we can see that here at lower substrate concentrations, rate of reaction is lower. And that's because if you have less substrate present, you're just less likely to have substrate present to collide with the enzyme active site, and therefore fewer enzyme substrate complexes, lower rate of reaction. So as you add more and more substrate, rate of reaction increases because you're going to have more successful collisions. So at this point, with the low substrate concentration, substrate's a limiting factor. But then we get this plateau in the rate of reaction, and therefore substrate concentration is no longer the limiting factor, and it must be something else. And in this case, it's going to be the enzyme concentration is now limiting, because if you add more and more substrate, eventually all of the enzymes are going to be saturated or in use. Every active site will have a substrate already bound to it. So that's why it levels off. For enzyme concentration, some similar concepts here, because at the low enzyme concentrations, you just don't have enough enzyme there to collide and bind with the substrate, so you have fewer enzyme substrate complexes. So at the lower enzyme concentrations, enzyme concentration is limiting the rate of reaction. But as you add more and more enzymes, then eventually you end up with enzymes with empty active sites, because unless you also add more substrate, then you're going to end up running out of substrate because that has been broken down or been turned into the product, and you just have more and more enzymes that are not in use. So it doesn't have any impact on the rate of reaction. Enzyme inhibitors then are molecules that can inhibit or prevent an enzyme from working. And you have competitive and non-competitive inhibitors. A competitive inhibitor has a very similar shape to the substrate. And because it's similar in shape to the substrate, that means it's almost complementary to the active site, and it can bind to the active site to form an enzyme inhibitor complex and prevent enzyme substrate complexes. And in this way, it reduces the rate of reaction. But if you add enough substrate to the reaction, that can actually knock out the inhibitor, and then the substrate can bind again. So that's why this one's known as competitive inhibitors, because they're both competing for the active site. Non-competitive inhibitors bind to the enzyme at a point that is not the active site, which is technically called an allosteric site, but you don't have to know that term to get the mark. You just need to know that it's a point that is not the active site. But when the inhibitor binds to that point, the allosteric site or not the active site section, it causes a change in the shape of the protein, so a slight change to the tertiary structure. And because we've slightly changed the tertiary structure, meaning that unique 3D shape, the active site changes shape, and therefore the substrate is no longer complementary and it cannot bind. So you still have this enzyme inhibitor complex, and you cannot form enzyme substrate complexes. And no matter how much extra substrate you add, that isn't going to reverse this. So this can be demonstrated with this graph here. We've got rate of reaction against substrate concentration. The blue line is showing us if there was no inhibitor, and that links to what we talked about previously with low substrate concentration, fewer collisions, but then it plateaus because you don't have enough enzymes. But in this one, we've now can see with both a competitive and non-competitive inhibitor, the rate of reaction is lower at almost all points until this bit here. And we can see that because the curve has shifted to the right. Now, the competitive inhibitor, when you get to the high substrate concentrations, it does actually have the same maximum rate of reaction when you have a high enough substrate concentration. And that's because the high concentration of substrate will knock the competitive inhibitors out of the active site. Enzymes can then all start working again, and therefore you get the same maximum rate of reaction. Whereas a non-competitive inhibitor, it doesn't matter how much extra substrate you add, it's going to be plateauing at a much lower rate of reaction because as that non-competitive inhibitor binds to the allosteric site, changes the shape of the active site, and therefore the enzymes can't work any faster because the active site isn't going to be in use. So this might be some of the enzymes haven't actually got a non-competitive inhibitor bound to them. That's why you get some rate of reaction still rather than 0. Next then, 3.1.5 nucleic acids. So deoxyribonucleic acid or DNA and ribonucleic acid RNA are important information carrying molecules. These two molecules will have the same function in any living cell that they're found in. DNA holds the genetic information, whereas RNA transfers the genetic information. Ribosomes are formed from RNA and proteins. That's such a small spec point that sometimes gets forgotten, but it's come up a couple of times now as a 1-mark question, so just remember that fact. And both DNA and RNA are polymers made up of the monomer nucleotides. So here we see a generic nucleotide. It has a phosphate group, a pentose sugar, and a nitrogen-containing organic base. But for DNA, you need to know that that pentose sugar is deoxyribose and that the options for the nitrogen-containing organic base are adenine, guanine, cytosine, and thymine. For RNA, the pentose sugar is ribose, and you still have adenine, guanine, and cytosine, but instead of thymine, you have the base uracil. So those are the two differences between the nucleotides, which pentose sugar it is, and thymine in DNA versus uracil in RNA. The polynucleotides then, both DNA and RNA form polynucleotides, which means a polymer made up of those nucleotides. And the way that they form is exactly the same as well. It's a series of condensation reactions, and you get a phosphodiester bond. That is the name of the bond that forms, and it's between the pentose sugar and the phosphate group. So for DNA, it's between the deoxyribose and the phosphate group. For RNA, it's between the ribose and the phosphate group. And this is a very strong covalent bond, which is important because we do not want this polynucleotide to break down, because the sequence of these bases is what is going to code for the sequence of amino acids when we come to creating proteins. So a few more things about this molecule then. A DNA molecule, so we're just focusing on DNA now, this is actually made up of 2 polynucleotide chains, and they join together by hydrogen bonds to form this shape here, which is a double helix. And you have complementary base pairs opposite each other, which is where the hydrogen bonds form. In contrast, an RNA polynucleotide molecule, that is just single-stranded, and they are much shorter compared to the length of a DNA polynucleotide molecule. So let's have a look then at this complementary base pairing. Paying close attention as well to the complementary spelled with an E, not an I. This type of complementary means like opposites attracting. Complementary with an I means saying something nice to someone. So we have our complementary bases here. Cytosine and guanine are complementary base pairs, meaning if they align opposite each other, hydrogen bonds can form. Adenine and thymine are the complementary bases as well in DNA. Adenine and thymine can form 2 hydrogen bonds, whereas guanine and cytosine can form 3. So this complementary base pairing is really important because it helps to maintain the order of the sequences of bases when DNA replicates, and also when mRNA is being created, to help ensure that the correct base sequence is maintained so that it codes for the correct amino acid sequence. So how the structure relates to the function then. So it's a very stable structure due to the phosphodiester bonds in the double helix. This is talking about DNA. The double strand means that when DNA replication occurs, both strands can act as a template. The hydrogen bonds between the two strands are quite weak, which is actually beneficial because it means they can be easily broken. So again, the two strands can separate and both act as a template during DNA replication. It's a large molecule, so it carries lots of genetic information, and those complementary base pairs enable identical copies of DNA to be made, which takes us on to DNA replication. So before any cell divides or replicates, whether that's by mitosis or meiosis, all the DNA in the cell must double or replicate first of all, so that when the cell divides, each new cell has its own copy of DNA. This process of DNA replication is described as being semi-conservative replication. What that means is, in the daughter DNA, which means the new molecule that's made, one strand is from the parental DNA, which means the original DNA molecule, and one strand is newly synthesized. So if we have a look here, this top bit is showing us the parental DNA, and we can see that we're going to make two new copies, and each of those has one strand of the original parental DNA, and one strand is going to be newly synthesized. That's what we mean by semi-conservative replication. So some key things then, before we go through the exact process. This is dependent on complementary base pairing. So again, just a reminder, it's adenine and thymine, cytosine and guanine, and there's two enzymes involved, DNA helicase, DNA polymerase, both of which you're commonly assessed on knowing what those enzymes do. So that's a key thing to make a note of here. Starting with DNA helicase. This is the enzyme that breaks the hydrogen bonds between complementary bases in the between those two polynucleotide strands. And that then causes the two strands to separate and also the double helix to unwind. Then within the nucleus, because this whole process happens in the nucleus, there are free floating DNA nucleotides that are attracted and align opposite to their complementary base pairs. Then DNA polymerase can catalyze the joining together of these adjacent nucleotides. So DNA polymerase is catalyzing the condensation reaction and the forming of the phosphodiester bond to create that new DNA polynucleotide chain. Now, the evidence for semi-conservative replication is linked to, first of all, Watson and Crick, who discovered the structure of DNA in 1953, but they were helped significantly by Rosalind Franklin's research on X-ray diffraction. And then they hypothesized that DNA must replicate either conservatively or semi-conservatively. And it was Michelson and Stahl who then conducted experiments to determine it is semi-conservative replication, not conservative. Now, these experiments I'm not actually going to explain in detail in this video, because this actually comes up on the spec saying as application questions, you need to be able to understand and explain it. But I do have a full video on this, which I'll link here, but just search Miss Estruch, Michelson and Stahl, and I've got a full explanation in detail of this experiment if you've come across questions on this that you weren't sure on. Right, next then, 3.1.6 ATP. ATP is a nucleotide derivative, and it is made up of an adenine base. So it's only got one nitrogenous base and it's always adenine. It has a pentose sugar, but it's ribose, and it has 3 phosphate groups. And its role is it's an immediate source of energy in metabolic processes. And all cells must have a constant, steady supply of ATP so that they can perform their metabolic processes. So let's go through some of the key details then. ATP hydrolysis, meaning the splitting apart of ATP, which breaks off an inorganic phosphate and releases energy. That is catalyzed by the enzyme ATP hydrolase, very similar to the word hydrolysis. So that reaction breaks ATP or hydrolyzes ATP into ADP, meaning diphosphate, T means triphosphate, and PI, which is inorganic phosphate. The energy released can actually be coupled to energy requiring reactions in cells, or that inorganic phosphate that's been broken off can be added to another compound, which is known as phosphorylation, and that then makes that new molecule more reactive. That happens at the start of respiration. Glucose gets phosphorylated, and you'll learn about that in topic 5. ATP can also be resynthesized, which is when ADP and PI join by a condensation reaction. It's also an example of phosphorylation because you're adding an inorganic phosphate onto this molecule. So this resynthesis is catalyzed by ATP synthase, and that happens in both photosynthesis and respiration, both which come up in topic 5. So here is our word equation. ATP plus water goes to ADP and PI. Energy is released, so it's not actually a product that you'd write in there because you can't produce or use up energy. 3.1.7 is water, and water is a polar molecule. One water molecule, so H2O, has unevenly distributed charge due to the fact that the oxygen is slightly negative and the hydrogen is slightly positive. So this symbol means delta, so delta negative, delta positive, which means slightly negative, slightly positive. And because of that, hydrogen bonds can form between the oxygen and hydrogen of different water molecules. And that concept underpins pretty much all of the properties we're about to see in water. So hydrogen bonds form between those different water molecules, like we just said, and there are 5 key properties of water that you need to know for AQA. I've put them here highlighted in yellow and what each one means. So it's a metabolite, which means it's used in many reactions. In this video alone, we've already seen it multiple times in condensation and hydrolysis reactions. In an exam, I would state that as well. Although it's in brackets here, I would say, for example, condensation and hydrolysis reactions. Number 2, it's an important solvent, meaning you can dissolve solutes in it. And that is really important because metabolic reactions happen in solution. Then it's got a high heat capacity, which means it takes a lot of energy to raise the temperature of water. And that's because of these hydrogen bonds. But in terms of in the exam, they just want you to say that means it buffers temperature changes. Number 4, it has a large latent heat of vaporization. You do have to get this the right way round. High heat, large latent. You can't say high latent. So think of the L's together. Large latent heat of vaporization. That means it takes a lot of energy to convert water in its liquid state to its gaseous state. And again, that's because of these hydrogen bonds. You'd have to have energy to break the bonds to then convert it from liquid to gas. So that means when you sweat, water provides a cooling effect because it takes a lot of energy to evaporate the water from your skin. And that energy comes from the heat in your skin. So it provides a cooling effect. And number 5, it has strong cohesion between water molecules. And that is because of the hydrogen bonds. Cohesion meaning like sticking together with those hydrogen bonds. So because water molecules form these hydrogen bonds between each other, they all stick together and you can get these continuous columns of water inside of plants, inside of the xylem, which comes up in the cohesion tension theory of topic 3. And also it provides surface tension where water meets air, meaning that you can actually balance some items on top of water, and that provides a habitat for organisms. Last bit of topic 1 is 3.1.8, inorganic ions. Inorganic ions are found in solution in the cytoplasm and in other body fluids. They're present in the body in varying concentrations depending on different ions, which they are. Each ion has a specific role, and most of these come up throughout the A level in different places. We've briefly talked about in this video the role of hydrogen ions, because we talked about that in terms of enzymes and how lots of H+ means that you have a more acidic solution or lower pH. So hydrogen ions affect pH. Iron ions comes up more in topic 3 when you learn about hemoglobin, because hemoglobin contains iron ions and is actually the part of hemoglobin that oxygen binds to, so it's involved in oxygen transport. Sodium ions come up in multiple places in the A level. The first one you're going to see is in topic 2 for co-transport, and then again in topic 3 because you have co-transport in absorption of digested molecules. So glucose and amino acids are transported from the ileum into the blood by co-transport. Phosphate answers come up in this video because they're components in DNA, RNA, and also ATP. AQA, a topic to the entire topic. Let's get into all of this information then, starting with 3.2.1, which is cell structure. For the eukaryotic cells, these are the 10 organelles that you need to know both the structure and how that structure links to the function. So we're going to go through all of these. Here's just showing you an overview though of what they look like inside of the cell. So we have our cell membrane on the outside. Now, although it does say vacuole here and it's an animal cells don't have permanent vacuoles, but they can have temporary vacuoles, things like your phagosome that you get in phagocytes, but we can kind of ignore that for the sake of this slide. Rough endoplasmic reticulum, so you've got these folded membranes with the ribosomes on the outside, so they look rough or bumpy. Golgi apparatus, the one that looks like a Wi-Fi symbol. The cytoplasm, lysosomes, ribosomes, which are the smallest, and then smooth endoplasmic reticulum and the mitochondria. So centrosome is not on the AQA spec. In plant cells, they have all of those structures plus 3 additional ones. They do have a permanent vacuole, which is surrounded by a membrane called a tonoplast. They have the chloroplasts, which is the site of photosynthesis, and then they also have the cellulose cell walls. So if we have a look at the nucleus first of all, the structures and the functions. The nucleus has got an envelope, which is this membrane around the outside, this double membrane, and there are holes within that called nuclear pores. The liquid in the middle, or the jelly-like material, is the nucleoplasm. You have the chromosomes within the nucleus, which are protein bound and described as being linear DNA. And then we've got the nucleolus, which is this additional small sphere inside of it, which in terms of the function, really that should be on that side, but the function is the site of RNA production, and it makes the ribosomes. So if you were asked to describe the functions of the nucleus, one of them we've already said down here, RNA production, and it makes ribosomes. It also contains all the genetic code for each of the cells, and it's the site of DNA replication and transcription, which is the making of mRNA. Endoplasmic reticulum can be rough or smooth, and both of them are made up of folded membranes called the cystinae. But the rough has ribosomes attached to the outside, which is what makes them rough. So here it is under the microscope, and all those tiny black dots are the ribosomes, giving that rough feel or rough look. The function of the rough endoplasmic reticulum is for protein synthesis, and the smooth endoplasmic reticulum is the synthesis of the lipids and carbohydrates. The Golgi apparatus and then the Golgi vesicles that pinch off. First of all, the structure though, it's folded membranes making these cystinae again, and it creates this Wi-Fi symbol is what I hear most people describe it as, because you've got these curved folded membranes and then the vesicles bubbling off. So those secretory vesicles pinch off from the cystinae. There's a whole range of functions for the Golgi apparatus and the vesicles. So you could have carbohydrates being added to proteins to form glycoproteins. You also get secretory enzymes being made, secrete carbohydrates. They transport, modify and store the lipids. They form lysosomes. Molecules are also labelled with their destination here, meaning you might add on some sort of chemical tag so it can only bind to a target cell. And also we've got the finished products are transported to the cell surface through these Golgi vesicles. They move up and fuse with the cell surface membrane, and that then releases the contents of the vesicle. The lysosomes are these bags of digestive enzymes. We can see one here. So just like sacs filled with digestive enzymes. So all of their functions are going to be to do with hydrolyzing and digesting. So they could be hydrolyzing pathogens inside of the phagosome. So in your phagocytes in topic 2, which we'll get into later on in this video, you've got your phagosome that has got a pathogen engulfed within it. The lysosome contains digestive enzymes, it fuses with the phagosome, and that digests the pathogens. It could be to completely break down dead cells within the body that you don't need anymore. You could have exocytosis, meaning the release of those digestive enzymes to outside of the cell to destroy material. And also it might be to digest worn out organelles so that you can reuse those materials. The mitochondria is a double membrane bound organelle. we've got the outer membrane here, and the inner membrane is folded. It's shown as this bright yellow structure here, folded so that you've got a really large surface area for some of the stages of respiration. So that folded inner membrane is called the cristae. Inside of that, you have a fluid-filled centre, which is known as the matrix. You learn more about this in topic 5 when you learn aerobic respiration. You also have loops of DNA and 70S ribosomes, which are the smaller ribosomes that you typically get in prokaryotic cells. And you need both of these structures so that within the mitochondria, you can have the creation of proteins, for example, enzymes that are needed for aerobic respiration. So the function then is it's the site of aerobic respiration, not anaerobic, just aerobic, which means you have lots of ATP being produced here, and also we get enzymes being created for respiration. Ribosomes, these are small, they're the smallest of the organelles, made up of 2 subunits of protein and RNA. Those are the 2 molecules that a ribosome is made up of. You have 2 different sizes, ATS, which are the larger ones, which are found in eukaryotic cells, and 70S are smaller in size, which are found in prokaryotic cells, but also inside of the mitochondria and chloroplasts in eukaryotic cells. And the function is for protein synthesis, meaning making proteins. Then if we have a look at a vacuole, so we're thinking about a permanent vacuole that you get in plants here. This structure is filled with fluid and it's surrounded by a single membrane, which we can see here as this darker layer, which is known as a tonoplast. The function is by having liquid in the centre, it pushes everything outwards and that makes the cell turgid and therefore it provides support. It can also be a temporary store of sugars and amino acids, and the pigments that may colour petals to attract pollinators can be found there as well. Next then, chloroplasts, which again are found in plants. This is very similar in structure to a mitochondria. It's also double membrane bound, but this time we have the outer membrane and the inner membrane directly next to each other, both creating a sort of oval shape. Instead, these folded stacks are additional membranes called thylakoids. So we've got these folded thylakoid membranes that fold up to create almost like pound coin or disc-shaped stacks. And those stacks are known as granum for singular or grana for plural. You learn more about that when you get on to topic 5 for photosynthesis. You also have a fluid-filled section here, which is known as the stroma. And the function is photosynthesis. Cell walls, which are found in plants and fungi, but also in bacteria, but we'll come back to bacteria in the prokaryotic cell section. So the cell wall in plants is made of cellulose. The cell wall in fungi, which are actually also eukaryotic cells, is made of chitin. And whatever it's made of, the function is the same. It provides structural strength to the cell. Plasma membranes, this is found in animals and plants. In fact, it's found in all living cells. So it's the phospholipid bilayer, which we can see here. We've got one layer of phospholipids a second. We've got the hydrophilic heads on the outside, hydrophobic tails pointing inwards. And within the plasma membrane, we also have proteins embedded. We have some carbohydrates on the outside as well. And you also have a lipid called cholesterol embedded within. And the function of the plasma membrane is to control what can enter and exit the cell. And this comes up again later in this video when we look at transport across membranes. So that's the eukaryotic cells and their organelles. Next, let's have a look at prokaryotic cells, for example, bacteria. And we can see a range of the organelles here. Some on here you don't need to know, but you do need to know that it has a plasma membrane, a cell wall. Sometimes it has a capsule. You don't need to know the pili for AQA. We've got a flagellum here, which some of them have, and some of them may have one or more plasmid. You do need to know that they have cytoplasm, ribosomes, and then the DNA is as a loop of DNA in the cytoplasm. So let's have a look at that information. The key differences then are between a prokaryotic cell and a eukaryotic cell. Prokaryotic cells are much smaller. In exams, they insist that you say much smaller, not just smaller. They don't have any of the organelles that have membranes, and we describe that as membrane-bound organelles. They do have ribosomes, just like eukaryotic cells, except they're smaller in size, they're 70S. They do not have a nucleus. Instead, their DNA is found as a single circular DNA molecule in the cytoplasm. They do have a cell wall, but it's made up of a glycoprotein, not cellulose, not chitin, and that glycoprotein is called murine. Some of them might also contain any of these three extra structures. They might have one or more plasmids, which are small loops of DNA that only have a few genes on them, often the genes for antibiotic resistance. Some of them have a capsule around the cell wall as well. This prevents the cell from desiccating, meaning drying out, and also it helps to cover the antigens to evade the white blood cells. And they might have one or more flagella, which is for movement or locomotion. Viruses, these are not classed as living, so they're non-living and they are acellular. This has come up as an exam question before to define what those two terms mean. So non-living, it's classed as not being alive because it doesn't do any metabolic reactions within the structure or within that particle. It cannot move, it cannot respire, it cannot replicate without using a host, and it has no means of getting nutrition. It's called acellular, meaning it's not a cell, basically, and it's not classed as a cell because it doesn't have a cell membrane. Viruses are even smaller than bacteria, and they only contain three structures. They contain the genetic material, capsid, and attachment protein. The genetic material is this here in the middle, and that can be DNA and it can be RNA. It'll be one or the other. That genetic material is surrounded by a protein shell called the capsid, not to be confused with the capsule that some bacteria have. So capsid is the protein shell that protects the genetic material. And then the only other structure that all the viruses have are these attachment proteins on the outside, and that's what they use to attach to receptors on host cells. Now we can see additional structures in this picture. We've also got an envelope, and some viruses do gain a lipid envelope by taking it from the host cell that they infect. They aren't able to produce their own one. So that is the three things that all virus particles contain. And that's what we call them, a virus particle, because it's not a cell. So next then is looking at how you can study cells and how this has evolved so that we've been able to understand these organelles that different cells have. And you can use a microscope, cell fractionation and ultracentrifugation. These are things that we're about to go through. The types of microscope, magnification calculation, calibration calculations linked to this idea of the eyepiece graticule, and then cell fractionation and ultracentrifugation. Starting with these two key definitions, knowing what's meant by magnification and resolution. Magnification of a microscope refers to how many times larger the image size is compared to the actual size of the object you are viewing. Resolution of a microscope is the minimum distance between two objects in which they can still be viewed as separate, and therefore you get a clear, crisp image still. The resolution of any microscope is determined by the wavelength of the energy source. So for an optical microscope, light, visible light is the energy source, so the wavelength of visible light is what determines the resolution. For an electron microscope, beams of electrons energy source, so the wavelength of electrons is what determines the resolution. So if we have a look then at some of these differences between optical, which are the light microscopes, and electron microscopes, which can be scanning or transmission. First then is what it is that creates the image. So for light or optical, it's a beam of light, which is condensed to a fine point using a glass lens. An electron microscope, it's a beam of electrons, which is condensed to a fine point using an electromagnet. Optical microscopes have a much poorer resolution, and that's because the wavelength of light is longer. Electron microscopes have a much higher resolution or resolving power, and that's because electrons have a shorter wavelength. Optical microscopes have a lower magnification, and electron microscopes have a higher magnification. Optical, you can get colour images, whereas you can only get black and white images with an electron microscope. You can then artificially add colour to that image using different Photoshop kind of programs, but with the actual microscope, you only get black and white. And then optical microscopes, you can view living samples, but you can't in an electron microscope. And that's because when you're preparing the sample, it all has to be inside of a vacuum, meaning there is no air. And that's because the air would absorb the electrons and then you wouldn't get an image. So it has to be a vacuum. But because it's in a vacuum with no air, that means you can't create samples with living things because they would die. So just to see an image here, we've got what we can see with an optical microscope. Because they have a much poorer resolution, that means that we can't see the small organelles in a cell. We can see the cell membrane, not in detail, but we can see it's there, cytoplasm and the nucleus and the chromosomes when they condense, but we can't see all of the organelles and we cannot see inside the organelles. But we do get a colour image and we can look at living cells. With an electron microscope, the electrons, as we said, can be absorbed by air, so it has to be prepared in a vacuum, which is why we can't have living samples and it's why it's black and white. A transmission electron microscope, these are the ones where the electrons transmit, meaning pass through the specimen. And that is why we have to have extremely thin slices of the specimen. They are stained and placed in the vacuum, and an electron gun produces a beam of electrons that passes through the specimen. Some parts of the specimen absorb the electrons and appear darker, so that's how we get these different grayscale, black and white parts of the image. So you end up with a 2D image, but you can see inside of the organelles. So this here is a chloroplast, and we can see the membrane, we can see the thylakoids, we can see those stacks, the grana, so you can see details internally. A scanning electron microscope, the specimen does not need to be thin because the electrons are not passing through the specimen. Instead, the electrons are beamed onto the surface and the electrons scatter back, and because they scatter back in different places because of the contours, you end up with these 3D style images. You can't see the internal structures because the electrons bounce back or they scatter back, but you can get an idea of the contours creating a 3D image. The math skill link to this then is magnification, and it's being able to calculate the magnification, or you might have to rearrange the formula to work out the actual size. So the key thing is knowing the formula. I always remember it as I am, so image equals actual size times magnification, and then I'd rearrange it if I needed to work out magnification. And you usually have to do unit conversions with this because most organelles, the actual size will be measured in micrometers because they're very, very small structures. But when you're measuring your image size, that is usually going to be in millimeters. So that means when you do your calculation, let's say we're working out the magnification and we're doing image size divided by actual, they both need to be in the same units. So you'd either need to convert your millimeter image size into micrometers, and to convert millimeters into micrometers, you would times by 1,000. Or you'd need to convert your actual size, which would be in micrometers, into millimeters, and that'd be divided by 1,000. Now you could also be asked to calculate the actual size, and in which case we need to swap actual and magnification here. So actual size would equal image size divided by magnification. And if they want you to work out actual size, they'll probably state that they want the answer in micrometers. So again, you might need to do a conversion. Then we move on to what is the eyepiece graticule. So inside of the eyepiece, which is this part here, there is a scale on a glass disc, which is called the eyepiece graticule. So when you look through the eyepiece, you will see what looks like a ruler. And this can be used to measure the size of objects that you are viewing underneath the microscope. But you need to know what each of these divisions on this graticule or ruler is worth. And that is going to vary depending on what magnification you are viewing the specimen at. So that's why we have to calibrate the eyepiece graticule every time we change the magnification. And when we say calibrate, what that means is work out what each of these divisions is worth in terms of a distance. So the way that we would do that is you have your eyepiece graticule in the eyepiece, but you also place a stage micrometer on the stage, which is this bit here. And a stage micrometer is typically 2 millimeters long and it has subdivisions 10 micrometers apart. So it's a glass slide that already has known distances etched onto it. So you line up your stage micrometer on the stage of the microscope, and you would maybe need to twist the eyepiece a little bit so that the eyepiece graticule is now completely lined up next to it. And we then need to look at how many divisions on the eyepiece graticule fit into one division on the stage micrometer. And we know that one division on the stage micrometer is worth 10 micrometers. And we can see that we've got 2 fit into one division. So that then means if that is worth 10 micrometers, we need to do 10 divided by 2, which is 5. And that tells us that each of these small divisions on the eyepiece graticule is worth 5 micrometers at that particular magnification. So then anything you view, you can use your eyepiece graticule to measure the size. And then we know that the number of divisions times by 5 is the actual size. And then if you're going to go up and change the lens to a different magnification, you would do this process again to work out what one of these divisions is worth on the eyepiece graticule. Next is cell fractionation, and this is used to isolate different organelles so that those organelles can then be studied. And this has helped us to identify organelle structures and functions. So there's 2 stages to this. We do homogenization and then ultracentrifugation. And homogenization, this is when the cells are broken open to release the contents, and the organelles are then separated by ultracentrifugation. But throughout this process, the cells must be in a particular prepared solution. It has to be cold, isotonic and buffered. And you need to be able to explain separately for all of these points why it has to be cold, then why it has to be isotonic, and then why it has to be buffered. So because you're breaking open the cell, that means you're going to have some organelles now exposed to enzymes that they might not usually be exposed to in the cell because we've broken it all open. And those enzymes might damage the organelle before we can examine them. So we make sure it's very cold, it's ice cold, to reduce enzyme activity, to prevent any enzymes potentially damaging the organelles before we can view them. It has to be isotonic, which means the water potential of the solution is the same as the water potential inside of the organelles. And this is to make sure that there is no osmosis, because we don't want any water moving into the organelles because that could cause them to burst. And we don't want water moving out because that could cause them to shrivel. And then it has to be buffered, meaning you use something to maintain the pH, because we don't want the pH to change because that could damage the organelles. And the key thing I've underlined each time is organelles. If you say damage the cell, that is incorrect because we've already broken open the cell. The cell doesn't exist anymore. It's the organelles that we need to now make sure they're undamaged so that we can go on to examine them. Okay, so a bit more about this homogenization process, basically like a blender. And in fact, when I've done this in lessons, that is literally what we used, a blender. So homogenization is when you break open the cells, and we do that with an ice cold isotonic buffered solution. Then you have to filter your solution to remove large cell debris. And then that filtered solution can be spun in a centrifuge at varying speeds, starting at low speeds. And this is going to separate organelles out from that solution according to their densities. So that's what we can see here. Here is our original solution that we've filtered. the centrifugal spin and the centrifugal forces cause pellets to form at the bottom of the most dense organelles. So the first time we spin it, we start at a low speed and then we increase the speed each time. But our lowest speed, that is going to cause the nuclei to sink to the bottom in the first pellet because the nuclei are the most dense. You then remove the pellet and you then put the rest of that liquid or the supernatant into another tube, spin it at a slightly higher speed. And this time you'd get the chloroplast if you're using plants and also the mitochondria because they've got similar density. Again, remove the pellets and the supernatant can be spun again. Next then it'd be lysosomes, after that endoplasmic reticulum, after that the ribosomes. And you do need to remember that order. They have done exam questions where they've asked, how would you isolate mitochondria? And for the mark, you needed to know it'd be on the second spin. So chloroplasts and mitochondria are both on the second. We then move on to 3.2.2. All cells arise from other cells, or in other words, we're looking at the cell cycle and mitosis. Now, not all cells are able to continually divide in multicellular organisms, but the ones that can go through the cell cycle. And in eukaryotic cells, when they enter the cell cycle, they will then divide by mitosis or meiosis. Prokaryotic cells replicate by binary fission, and viruses do not undergo cell division because they are acellular, meaning they don't have a cell to divide, and they are non-living. Viruses can still replicate though, but only once they are inside of a host cell, which is the cell that they are infecting. And they have to invade by injecting their nucleic acid into the cell to replicate the virus particle there. And that comes up later in this topic in immunology. So binary fission then, we said this is how prokaryotic cells or bacteria replicate. And this involves, first of all, we have the circular DNA and the plasmids, if they have plasmids, will replicate. And that's what we can see here. We've got circular DNA and we've got plasmids. That is going to replicate. The cytoplasm splits to then create two daughter cells. And each daughter cell has one copy of the circular DNA, but the plasmids, if they are present, don't necessarily split equally, so they might get different numbers of plasmids. It's shown here that they have the same, but it could be different. So then let's have a look at the cell cycle. There are three key stages: interphase, nuclear division, and cytokinesis. Interphase is the longest stage, and we can see that here. Interphase is shown here as G1, S phase, and G2. So we can see about 90% of the cell cycle is interphase. And this is when the organelles double, the whole cell grows, and also DNA replication occurs. So G1 of interphase, that is the growth phase. So that is when we're going to have the growth of the cell and also some of the organelles doubling. S phase is when the DNA replicates. And G2, we have more growth. Also, we have checks for any DNA replication copying errors, and we have final preparations for mitosis, or it could be meiosis. And then this stage is our nuclear division, which can either be mitosis or meiosis. In this topic, topic 2, we only look at mitosis, and that is when you create identical diploid cells. Diploid, di meaning two. Diploid cells are ones that have two copies of the chromosomes. Meiosis, which you learn in topic 4, you end up with 4 genetically different daughter cells are haploid. Haploid meaning one copy of each chromosome. Mitosis creates cells with identical DNA, and therefore it's useful for growth and repair, whereas meiosis creates gametes, which are all genetically different. The final stage, which is just shown as this red line here, it's a very, very short stage, is cytokinesis. And this is when we get the division of the cytoplasm and the cell membrane to create the new cells. So we're going to focus on mitosis, which is just one of these stages in the cell cycle. And mitosis is split up into multiple steps as well. We have prophase, metaphase, anaphase, and telophase, or PMAT, to remember the order. Key facts about mitosis: you only have one division occurring, so you go from having one parent cell to two daughter cells. You get identical, genetically identical cells. They are diploid, meaning two copies of each chromosome. We've only got one chromosome shown here, but we can see we've got the red copy and the blue copy. That's in the parent cell, and it's the same in the two daughter cells. And as we said, the function, growth and repair, and we're going to see that later in this topic when we see clonal expansion in immunity. So prophase, this is our first stage. What you need to know throughout mitosis is what is happening to the chromosomes. In the spec, they describe it along the lines of the behaviour of the chromosomes. So that's what we're going to focus on. What's happening to the chromosomes and why? So in prophase, the chromosomes condense and that makes them visible. So that is what is happening. And in animal cells, these centrioles will also move to be at the opposite poles, which means opposite ends. The centrioles are responsible for creating the spindle fibres, and because they move to both ends, they start to releasing it from both poles, and they'll start to attach to the centromere on chromosomes and the chromatids on the chromosome in later stages as well. Plants have spindle apparatus, but they don't actually have centrioles. Metaphase. So in terms of what is happening to the chromosomes will now line up in the middle of the cell, which is known as the equator. Spindle fibre that is released from the centrioles will now attach to the centromere of these chromosomes, and that's what causes them to go in this single file line at the equator. In anaphase, the behaviour of the chromosomes is now that they'll be split apart at the centromere, and then each chromatid is pulled to the opposite poles of the cells. So those spindle fibres that were produced start to retract, and that is what pulls and splits or divides the centromere in two, so that those individual chromatids are pulled to the opposite poles of the cells, so they're now separated. And once they are separated, we can now call those chromatids, once they're at the opposite poles, chromosomes again. This stage does require energy in the form of ATP, which is provided by respiration in the mitochondria. Telophase is our last stage. The chromosomes are now at each pole of the cell and they're going to uncondense. So they become longer, thinner and no longer visible. Spindle fibres will start to disintegrate, the nucleus starts to reform, and then the very last step, which is not in mitosis but it is in the cell cycle, is cytokinesis, which is when the cytoplasm splits, so does the cell membrane, so you end up with two genetically identical daughter cells. You could be asked to calculate the mitotic index, and this is a way of looking at the proportion of cells that are currently undergoing mitosis compared to the number of cells present when you're looking at a sample underneath a microscope slide, which links to required practical too. So in order to calculate the mitotic index, in your field of view, which means the section that you can see when you look through a microscope, you need to count how many cells are currently in mitosis. The way that you can tell that is whether you can visibly see chromosomes, which is squiggly lines. If it just looks like grey, or in this case it's stained purple, if it just looks like dots almost and you can't see any squiggly lines, that's an interphase, so that is not in mitosis. And most cells, if you remember, we said 90% of the cell cycle is interphase, which should mean about 90% of the cells will be in interphase, and only about 10% will be currently going through mitosis. So we'd count how many cells are in mitosis, divide that by the total number of cells present, and that gives you a mitotic index. You might get questions on how can you standardize this, so you could say you're only going to count cells in which you can see the entire cell in the field of view. Now, uncontrolled cell division comes up again later when you learn more details about cancer in topic 8. But mitosis is a highly regulated process, meaning it's very controlled to make sure that cell division is accurate and it's only happening when it's needed. But disruptions to this control do occur, and that then results in uncontrolled cell division, which means you're going to get lots and lots of cells being made, even if you might not need them. And that is how you get a tumour forming, which is just a mass of cells. And those tumours could be benign, which means they're not cancerous, or malignant, which means they are cancerous. And because these tumours are formed because of the uncontrolled cell division of mitosis, many cancer treatments will target mitosis to try and control the rate. Now, this mainly comes up as application questions for topic 4. You learn more theory on this in topic 8 in year 13. 3.2.3 is the next part of topic 2, transport across cell membranes. So this takes us back to the plasma membrane, the cell membrane, which could be a cell surface membrane, but also an organelle membrane. So when we talked about the chloroplast being a double membrane bound organelle, they have one of these membranes. Well, they have two of them because it's double membrane bound. So the membrane is described as a fluid mosaic model, and that's due to the fact that it's a mixture of different molecules. That's why it's a mosaic. So it's made up of the phospholipid bilayer. We've got proteins embedded within it, but also just pinching in externally. We have glycoproteins and glycolipids, and it's described as fluid because there is some slight lateral movement of those phospholipids. All of these molecules are arranged within that phospholipid bilayer, and that is what creates the partially permeable membrane, which is that property that we talked about, or the function. It can select or control what can pass through the cell surface membrane. So let's just go through this in a bit more detail. The main component of the membrane is phospholipids, and this links back to topic 1, which if you haven't already seen my entire topic 1 video where I go through lipids, or if you haven't seen my lipids video in detail, then just search Miss Estruch lipids or Miss Estruch phospholipid bilayer, Miss Estruch entire topic 1, you'll find it. So the phospholipids align as a bilayer, bi meaning two, so 2 layers, that's what it means. And we have the hydrophilic heads on the outside, the hydrophobic tails are on the inside. This is actually zooming in on one phospholipid, and we've got these structures here that need labelling. So 1 is the phosphate group, 2 is the glycerol, 3 and 4 are both fatty acids, but 3 is a saturated fatty acid, 4 is an unsaturated fatty acid because of that double bond between the 2 carbons. A is the hydrophilic head. Hydrophilic because of this negative charge on the phosphate group. And then B is the hydrophobic tail. Those fatty acids are non-polar, so they are hydrophobic, meaning they will repel water, whereas hydrophilic, hydro meaning water, hydrophilic heads will interact with water. So that's why phospholipids in aqueous solution or aqueous environments will create this bilayer. The heads can interact with any of that solution, the water-based solution on the outside, but it will repel the tail, so they spin inwards, and that's how you get this bilayer. Now this goes towards making this partially permeable membrane property because molecules can only move through the phospholipid bilayer by simple diffusion if they can dissolve in lipids and if they're small enough. So it has to be a lipid-soluble substance, which means it has to be non-polar, so it can't be charged. And that's going to be some hormones like estrogen, and also very small molecules like carbon dioxide and oxygen gas are able to. Molecules that can't pass through the membrane, and by that I mean dissolve through the phospholipid bilayer by simple diffusion, is anything that is polar or water soluble, so it has a charge, because that will be repelled by those phospholipids. So things like sodium ions. Or any molecule that is too large to fit through the gaps, so glucose for example. Now you do have other components in the cell membrane, making that mosaic feature of the fluid mosaic model. So cholesterol is another type of lipid, and that's present in some membranes in varying amounts. And cholesterol restricts the lateral movement, meaning the sideways movement of other molecules in the membrane. So that's going to be useful as it makes the membrane less fluid at high temperatures, and we want that so that it can prevent water and dissolved ions from leaking out. You also have proteins in your membrane, and proteins can be embedded across the cell surface membrane, or they could be just on the outside. So if it's embedded across, it's known as integral. If it's just on the outside, it's known as peripheral. Sometimes that's intrinsic and extrinsic. So the peripheral proteins, those can provide mechanical support, or they can connect to carbohydrates to make glycoproteins, and carbohydrates can bind to the lipids to make glycolipids. And those are normally, the function is for cell recognition as receptors. The integral proteins are going to be carrier proteins and channel proteins, which are involved in active transport, co-transport, and facilitated diffusion, which is coming up shortly. Protein channels, which we can see one here, these form tubes. We can see like a hollow-like tube, and that fills with water. That will enable any of our polar substances, so they're going to be water soluble, to dissolve in that water, and then they can move through the membrane in that way because they can't dissolve in the phospholipid bilayer. Whereas carrier proteins, these will have a molecule bind with them, such as glucose or amino acids, and when that molecule binds to the carrier protein, it causes it to change shape, and that then releases it on the other side of the membrane. Which leads us into the 5 key ways that molecules can transport across these plasma membranes. We have the top 3, which don't require energy: simple diffusion, facilitated diffusion, and osmosis. And the bottom 2 are both active types of transport, meaning they require energy from ATP from respiration. Active transport and co-transport. So let's go through all 5 of these. Now, one thing that they'll all have in common is there are certain adaptations to increase the rate at which those types of transport happen. So you could be asked to identify adaptations that cells have for rapid transport. And the sorts of things you need to look for are features that increase the surface area. So for example, microvilli, which is highly folded membranes, increases the surface area. Or if it's a type of transport that goes through a protein channel or a carrier protein, then there might be more of those proteins embedded in the membrane. So again, you've got a larger surface area for that to happen. If it's a cell where there'll be a lot of active transport or co-transport, because those require ATP from respiration, you'll also have many mitochondria as an adaptation as well. So let's have a look at simple diffusion. This is the net movement of molecules from an area of higher concentration to an area of lower concentration. So it's going down the concentration gradient. And that will occur until equilibrium is reached, meaning same concentration in both sides. This process doesn't require any additional energy from ATP. The molecules do still have energy because they're moving, but this is due to kinetic energy that they possess, and that's what enables them to constantly move in fluids, whether that's gas or liquid. For small molecules to diffuse across a membrane, they must be lipid soluble and small. So that's the criteria for simple diffusion across a membrane. It has to be lipid soluble, so that would be a non-polar molecule, meaning it doesn't have a charge, or a small molecule. Facilitated diffusion is still going down the concentration gradient. So it's still a passive process. No extra energy is added in the form of ATP. It's going down the concentration gradient, but it's for any molecule that doesn't fit the criteria we said here, lipid soluble and small. So it's going to be for molecules that are polar, so that means they have a charge, they are not lipid soluble, they're water soluble, and they are too big, or they might be too big. So this is going to happen through either protein channels or protein carriers. It can be through either of those types of membrane proteins for transport. If it's a protein channel, this is the ones where we have the tube-like center filled with water. That enables the ions or polar molecules, water-soluble molecules, to dissolve and then pass through the membrane through that tube filled with water, that protein channel. It is still selective though, as the channel proteins will only open in the presence of certain ions when they bind to the protein. So it is still selective transport across the membrane. Facilitated diffusion can also happen with carrier proteins, and in this case the molecule will bind to the carrier protein, such as it could be glucose, and that causes a change in shape to that carrier protein, and that change in shape causes it to release the molecule to the other side of the membrane. Osmosis, so this is still passive, meaning no extra ATP or energy is needed, and it is going down a concentration gradient. But we need to use the terminology water potential, not concentration gradient, for osmosis to get the marks. So for this, water potential is the pressure created by water molecules, and it's measured in kilopascals and represented by this symbol here. This means water pressure. Pure water has a water potential of 0. So 0 is the highest water potential you can ever get, and that's for pure distilled water. As soon as you dissolve any solutes in it, you get a more negative, or you get a negative water potential. And the more negative a water potential is, that indicates more solutes are dissolved in it. So that's what we mean by water potential. And osmosis is the net movement of water from an area of higher water potential, even though it will still be negative unless it's pure water, but it's a higher water potential to a more negative or a lower water potential, and that's the direction that the water will move, and it's across a partially permeable membrane. So water is the thing that moves, not the solute, and it's always moving towards a more negative water potential, which means more solutes are dissolved in the water at that location. Now, you could get questions linked to this, or mainly actually links to the required practical to do with plant tissue, but the terms isotonic, hypotonic, and hypertonic. Isotonic means the water potential of a solution is the same as the water potential in a cell. And because there's no difference in the water potential, you don't have any osmosis happening, or there's no net movement of water, so the cell will stay exactly the same size and shape. A hypotonic solution is when the water potential of the solution is more positive, meaning closer to 0 than the cell. And that means there's more water in the solution than the cell. So water is going to move into the cell by osmosis. And in animal cells, that can cause it to swell and even burst. Hypertonic solutions is when the water potential of the solution is more negative than the cell, and that will cause water to leave the cell by osmosis, and the cell will then shrivel up. Next, then we move on to our active types of transport. So we have active transport first, and this is the movement of a substance from a low concentration to a high concentration. So it's going against the concentration gradient. To do this, it has to use metabolic energy, so that means ATP from respiration, and also it always goes through a carrier protein. So you have to state it's a carrier protein for active transport. So the way that this happens then is, step number 1 is the transport is going through, or transport is occurring with a carrier protein, and those are ones that are going to span the entire cell membrane. A molecule will bind to a receptor on that protein, complementary in shape. ATP will then also bind to the carrier protein from the inside of the cell. So we can see here in the cytoplasm on the inside, it then binds, and it's hydrolyzed into ADP and PI. That releases energy, and that is what causes the carrier protein to change shape and release the molecule to the other side of the membrane. This inorganic phosphate ion was attached the whole time after ATP has been hydrolyzed, and that then gets released, and that causes the protein to return to its original shape. So in order for active transport to continue to occur, you need a constant supply of ATP from respiration. Next then is co-transport, and this is a type of active transport because one of the steps in the process is active transport. So to absorb glucose from the ileum lumen in the gut, which we'll see in topic 3 in digestion and absorption, because there's a higher concentration of glucose in the cell than in the ileum, it's hard to get the glucose to move in because it's going against a concentration gradient. So that's why we need a type of active transport, which is co-transport. So the first thing that happens is actually here. Sodium ions from within a cell are actively transported out of the cell and into the blood in capillaries. That then reduces the concentration of sodium ions in the cell. And as a result, we have now created a concentration gradient from the ileum to the cell. So sodium ions are able to move from the ileum into the cell down a concentration gradient. But they are an ion, they're sodium ions, so they have to do this through a protein. And the protein that they do this through is a co-transporter protein, which means it transports 2 molecules at the same time. And it transports the sodium ions plus glucose, or it could be amino acid as well. That then gets the glucose into the cell. And then we have a high concentration of glucose in the cell compared to in the blood. So glucose can now move down its concentration gradient from the epithelial cell into the blood and the capillary by facilitated diffusion through a protein. And it has to be facilitated because glucose is too large to move by simple diffusion. Next then is 3.2.3, cell recognition and the immune system. So the first part is all about cell recognition and identifying what are self cells, meaning part of your body, and non-self cells, which are not part of your body. And your immune system has cells to identify the presence of pathogens, which are non-self cells, and other non-self cells, which are potentially harmful. And then your immune system will either destroy those or neutralize them to prevent harm. So those cells are the lymphocytes, and each type of cell has a specific molecule on its surface to identify it. And those molecules are usually proteins, and because they're proteins, they have a unique 3D shape. There's overlap here with biological molecules from topic 1 because common marking points linked to this are stating that it has a tertiary structure because it's a protein and that's what gives a unique 3D shape, but tertiary structure would be the key marking point, and that would be complementary in shape to, and that's actually going to link to some of the cells that we learn about later, but it's the idea of tertiary structure, complementary in shape. That's how you can work out if it's a self or non-self cell, and a bit more on that when we get to the T and B cells. But thinking now more about what counts as a non-self cell. So if a non-self cell is detected, a response will be triggered to destroy that cell. And these are the 4 examples that you need to know that could trigger an immune response because they're detected or could be detected as non-self cells. So number 1 is a pathogen. So bacteria, fungi, they'll have antigens on their surface that can be used to identify them as non-self cells. It could be cells from another organism of the same species. So for example, if you've had an organ transplant, that could be detected as non-self. And we don't want that to happen because then your immune system will start to attack and destroy the cells from that transplanted organ. It could be abnormal body cells. So for example, cancer cells, their antigens on the surface will slightly change. They now don't get detected as self cells, they're now detected as non-self. Or it could be toxins because some pathogens release toxins into the blood, such as cholera. So I use this word antigen quite a bit, but an antigen is a foreign protein that generates an immune response by the lymphocytes when it's detected, and it's located on the surface of cells. That is quite a common 2-mark question. You need both of those points to define what an antigen is. Antigens can change, though, over time, and this links to antigen variability. So a pathogen's DNA can mutate frequently, and if the mutation occurs in the gene that codes for the shape of the antigen, then that shape of the antigen will change. And therefore, any previous immunity that you might have had for that pathogen, based on the recognition of the old shaped antigen, it will no longer be effective because the memory cells have a memory of the old antigen shape. So this is known as antigen variability. And for example, the influenza virus, which is the virus that results in the flu, that mutates and changes its antigens very quickly. And that's why you need a new flu vaccine every single year. So let's go on to the immune response then. If you do have this non-self cell detected, we said an immune response occurs. So if a pathogen gets past any chemical or physical barriers, which you don't learn for AQA for A level, but that's things like your skin barrier, your stomach acid, and it can then enter the blood, then this is when the white blood cells are the second line of defense, and you either get a specific response by the lymphocytes or a non-specific response. And in fact, you get both, starting with the non-specific response of the phagocytes and then the lymphocytes. So the non-specific response by the phagocytes is phagocytosis. And a phagocyte is a type of white blood cell. For example, a macrophage is an example of a phagocyte. And phagocytes are white blood cells that can do phagocytosis. They're found in the blood, but also in the tissue. And phagocytosis is a non-specific immune response. What that means is any non-self cell that is detected, and that means any of those 4 items that we listed a couple of slides ago, if they are detected, it will trigger phagocytosis to happen every single time. So phagocytosis then, the first thing that happens is our phagocyte, for example a macrophage, which is in the blood or in the tissues, and any chemicals or debris that are released by the pathogen will attract the phagocyte to move towards the pathogen. So that's how the phagocyte is able to detect where the pathogen is. Now on the phagocyte, there are many receptors which are binding points on the surface of that phagocyte that can then bind to the antigens or any chemicals on the pathogen. The phagocyte, once it's then bound to it, the phagocyte changes shape, which is known as engulfing. So it completely surrounds and engulfs the pathogen, and it then will put it inside of a vesicle inside of the cell. And that vesicle is known as a phagosome. So we can see here the pathogen's been engulfed and it's now trapped inside of the phagosome. You then have the lysosomes within the phagocyte will move towards, so the lysosome shown here in blue, it will move towards and fuse with the phagosome, and it will then release its contents. And the contents of a lysosome is digestive enzymes. And in this case, they're known as lysozyme enzymes. They're digestive, which means they're lytic, or they're going to split, they're going to break apart, hydrolyze the pathogen. So that is how the pathogen is then destroyed. From the broken down, hydrolyzed, digested pathogen, any soluble products that are going to be useful can be absorbed by the phagocytes, and then the antigen is placed on the surface of the pathogen, and that is going to be used later on as a way to signal to other white blood cells known as lymphocytes that a pathogen has been detected and destroyed. So that takes us on to the first type of lymphocyte we're going to go through, T lymphocytes, or T cells for short. These lymphocytes are involved in the specific immune response, meaning they respond in a particular way based on the particular shaped antigens that are detected. Lymphocytes are made in the bone marrow, and this is extra information, you don't need to know it, but they're called T lymphocytes because T cells then go to mature in the thymus, which is this structure here in the body. And they are involved in the cell-mediated response. So this is where we look at how the antigen-presenting cells are going to help trigger the cell-mediated response. And the antigen-presenting cells could be, like we just said, a phagocyte, for example, a macrophage, which has engulfed and destroyed a pathogen and then it presents it on its surface. But it's actually any cell that has a non-self antigen on its surface. So that could be one of your own body cells that's become infected. It could be one of those phagocytes presenting the antigen on the surface. It could be cells of a transplanted organ. It could be cancer cells. And because they have antigens presented on their surface, that is going to enable lymphocytes to respond to those antigens in this cell-mediated response. So let's carry on from this bit we were at. So once the pathogen has been engulfed and destroyed by the phagocyte, antigens are then presented on the cell surface. It's now called an antigen presenting cell. And we can see an example of that here. We've got an antigen presenting cell. One type of T lymphocyte is known as a helper T cell. And helper T cells, they have these receptors on their surface, and that can attach to complementary shaped antigens on antigen presenting cells. Once it then attaches, that activates the helper T cells to divide by mitosis, and that will then replicate and make large numbers of clones of that exact helper T cell that had the receptor complementary in shape to that exact antigen. That stage is known as clonal expansion. Clonal because you're making identical copies, expansion because you're making large numbers. And this is where there's that link back to mitosis. We said the role of mitosis is growth and repair. In this case, it's growth and making large numbers of identical T helper cells. Now we've got that large number of identical T helper cells, they actually differentiate into different types of T lymphocytes. So some will actually just remain as helper T cells because those are then going to go on to activate the B lymphocytes, which we're going to go on to shortly. Some of them are able to stimulate macrophages so that they will perform more phagocytosis and engulf and destroy more of those non-self cells. And then some of them become cytotoxic T cells, sometimes called killer T cells. AQA use the term cytotoxic T cells. And a cytotoxic T cell is able to destroy your own body cells that are infected with a pathogen, typically a virus. And the way that they do this is cytotoxic T cells release a protein called perforin, and that protein can embed in the cell surface membrane to make a hole or a pore. And that means that either lots of water will move in and cause the cell to burst, destroying the infected body cell, or it will cause lots of water to move out, causing the cell to shrivel, and again, it will cause cell death. So this is most common in viral infections because viruses can only replicate when they're inside of the body cell. So if you destroy the body cell, the virus can't replicate, and that gives more time then for the phagocytes to locate and destroy the virus before it can replicate and cause more damage. And this is why you get a sore throat when you have a cold. The cytotoxic T cells are destroying the infected body cells in your throat. You're sacrificing body cells to prevent the virus replicating and infecting even more cells and tissues. Now, B lymphocytes, this is the humoral response. Again, all lymphocytes are made in the bone marrow. You don't need to know this, but the B is because it matures in the bone marrow as well. This is a humoral response, meaning within liquids or within body fluids, because in this instance, the B cells will only respond to antigens that are floating within the blood or within body fluids. They don't respond to the antigen presenting cells in the same way as a cell-mediated response with the T cells. So let's have a look at the B cell activation. So you have approximately 10 million different B cells which have antibodies on their surface, complementary in shape to 10 million different antigens. And you have these circulating in your blood, and if you get an antigen in your blood, collide with the complementary shaped antibody on a B cell, that is going to cause the B cell to take in that antigen by endocytosis, which means basically surround and engulf it. So this is responding to the antigens in the blood, not the entire cell. When the B cell then collides with the helper T cells that we talked about in the previous slide, that activates the B cell to go through clonal expansion and differentiation. Now this can happen because when that B cell takes in the antigen, it's going to break it down, but it can then actually also present it on its surface. So that means it's able to bind to helper T cells that have that receptor complementary in shape to the antigen. That is then going to trigger the B cell to be activated to do the same thing as the T cells. It undergoes mitosis, you get a large number of clones, clones of that B cell that has that exact shape, antigen and antibody on its surface complementary to that foreign protein, the antigen. But this time the cells differentiate into different molecules. They can either become plasma cells or memory B cells. The plasma cells, which you can see here, these are the ones that make antibodies. Memory B cells are the ones that remain in your blood for long periods of time, and they can divide rapidly into the plasma cells to release large quantities of antibodies so quickly if you get reinfected with the same pathogen. So they're responsible for you being immune, basically, to particular pathogens and disease. So let's have a look at this. Memory B cells, as I said, they can live in your blood for a long time. They can be decades in your blood, whereas plasma cells are very short-lived. And those are the ones that make the antibodies, so that's why we need to store a memory of that antibody. So if you are reinfected, you can then have the memory B cell rapidly differentiate into plasma cells, which can then make lots of antibody so quickly that it should help to destroy the pathogen before you get any symptoms, which means you are immune. Or it might just mean you get very, very minimal symptoms. And this is known as active immunity. You are immune to that pathogen, and it's active because you have memory B cells against that pathogen. So this links this concept of primary and secondary response. Primary immune response is the first time you are exposed to a pathogen. And because it's the first time you're exposed, it can take a longer period of time for the B cell to collide with that antigen, causing it to engulf the antigen, and then you get the clonal expansion, differentiation. All of that process of the humoral response has to happen. So it takes a longer time to make antibodies, and you don't get higher concentration. The second time you're exposed to that same antigen, because you now have the memory B cells present in your blood, those can then collide with the pathogen's antigen again and differentiate rapidly into plasma cells. So you end up with large quantities of antibody much, much quicker. And this is why the secondary response should mean that you get no symptoms or very few, because you get the antibodies being produced so rapidly and in large concentrations. So let's have a look at what antibody is. They are proteins that have a quaternary structure, meaning more than one polypeptide chain, and they happen to have 4 polypeptide chains. The end bits here are the variable regions, meaning those are the parts that are going to be different in shape and combine to the different shaped antigens. The red part is constant, meaning it's the same in all antibodies. The heavy chain is this longer one, and you have 2 heavy chains. The shorter chain is known as the light chain. Here are the antigen binding sites. You can bind one here and here. And then you also have these bonds here and the hinged section, which makes the antibody flexible, so it can easily bend and attach to one antigen over here, bend and attach to another over there, which is how we get agglutination happening. And agglutination is the idea that multiple antibodies combine together lots of different antigens at the same time, so they all clump together. That makes it easier for the phagocytes to, first of all, locate where all the pathogens are, and then they can more efficiently engulf and destroy them. So antibodies, they create these antigen-antibody complexes, and we get agglutination happening. But they don't directly kill the pathogen. They just make it far more efficient for the phagocytes to then destroy the pathogen. So different types of immunity. First of all, passive immunity. Passive immunity is when the antibodies are introduced into the body and the pathogen doesn't enter itself. So you don't actually get any antigen or pathogen in you. So that means the plasma cells and memory cells are not made. You just get a direct source of antibody. So you don't get any long-term immunity because there's no memory B cells. A natural example of this is in breast milk. So through breastfeeding, breast milk contains antibodies. That means babies will have passive immunity whilst they are breastfeeding. And when I say whilst, it's not literally just while they're breastfeeding. The antibodies can last for a short period of time. Active immunity is when you have been exposed to the pathogen or the antigen, and therefore you do have memory B cells in the body. And that could be natural active immunity following infection, or it could be artificial active immunity following a vaccination where you might have a weakened version of the pathogen or the antigens injected in. So vaccines, that means a small amount of either weakened or dead pathogen or the antigens itself introduced to the body. And that could be as a liquid through the nose or mouth, or it could be an injection. And exposure to those antigens activates the B cells to go through clonal expansion and differentiation, just as we saw in the humoral response. So those B cells will then undergo mitosis, make large numbers of identical cells, which will then differentiate into plasma cells and memory B cells. Plasma cells will make antibodies, which will then destroy any of that weakened or dead form of the pathogen. And the B memory cells will then remain in your body for decades. So if you are reinfected with the same pathogen, you can make large numbers of the antibodies so rapidly that you shouldn't get symptoms. Herd immunity is if enough of the population are vaccinated against a particular pathogen. That means that the pathogen cannot spread or transmit amongst the population. And that's because if you have most of the population immunized, that means that they can't transmit the pathogen onto someone else, and therefore hardly anyone in the population is going to have the pathogen. And this provides protection for those who are not vaccinated. So maybe they are too ill to be able to be vaccinated, or maybe they're pregnant, or they have some lowered immunity that means they aren't able to. The next bit is then looking at HIV, an example of a virus, and it is a retrovirus. It has a lipid envelope around it, but that is taken from the host cell membrane. The envelope has the attachment proteins, which are glycoproteins, which is how it attaches to the host cell. So here are the key structures that you have in the HIV particle. You have the core, and that is the genetic material, which is RNA, and it has the enzyme reverse transcriptase as well, which is needed for the virus to replicate. You have the capsid, which is the outer protein coat protecting the genetic material. There is the envelope, that is an extra outer layer, but that is made out of lipids taken from the host's cell membrane. And then you've got the protein attachments on the exterior of the envelope to enable the virus to attach to the host's helper T cell. So let's have a look at how HIV replicates in helper T cells, because those are the host cells that it infects. HIV is transported around in the blood until it attaches to a CD4 protein on a helper T cell. So here is a helper T cell. Here is a CD4 protein, which is a receptor. And we can see that the HIV with the attachment proteins binds to that receptor. The HIV protein then fuses with the helper T cell membrane, and that enables the RNA and the reverse transcriptase enzyme to enter with the capsid, and then it does get released from the capsid. The HIV enzyme reverse transcriptase copies the viral RNA into a DNA copy and moves into the helper T cell nucleus, and this is why it's called a retrovirus, because it's doing the reverse of transcription. You've got an RNA copy being turned back into DNA. The DNA then moves into the nucleus. It combines with the host's DNA, and that is where the mRNA is then transcribed along with the host cell's mRNA to create the viral proteins. The viral proteins are then reassembled, and they'll then move towards the cell surface membrane, and they are released from the cell. And that then goes on to make a new virus particle. Now, the difference between HIV and AIDS is HIV is the virus, and it specifically targets and it destroys helper T cells, because as it replicates inside of them, it destroys the helper T cells, which activates B cells. So if we don't have those helper T cells, they're not there to activate B cells. That means we have now significantly reduced the immune system. And if enough helper T cells are destroyed, then you end up with AIDS, acquired immunodeficiency syndrome, where you've become so vulnerable to opportunistic infections and cancers and you can't defend yourself. So symptoms of AIDS are chronic infections, weight loss, fever, night sweats, and they can be followed by more serious illnesses like pneumonia, tuberculosis, and certain cancers. A person is HIV positive if they're infected with HIV. AIDS is when the replicating virus in the helper T cells interfere with the normal function of the immune system, and it's normally classed by how many of the helper T cells have been destroyed. The last bit of this topic is monoclonal antibodies. Mono meaning one, clonal meaning identical. And a monoclonal antibody is a single type of antibody that can be isolated and cloned. And antibodies are proteins which have binding sites complementary in shape to certain antigens. Now this is what is created in the immune response, monoclonal antibodies, lots of identical copies of the same antibody. But this has been manipulated to create monoclonal antibodies for medical treatments, medical diagnosis, and even pregnancy tests and other chemical tests. Now, targeted medication, this can use direct monoclonal antibody therapy, and this is when you have some cancers that can be treated using monoclonal antibodies, which are designed with a binding site complementary in shape to the antigens on the outside of cancer cells. Those antibodies are then given to the cancer patients and they'll attach to the cancer cells. While the antibodies are bound to the cancer cell antigens, this prevents chemicals from binding to the cancer cells, which enable the uncontrolled cell division. And therefore, the monoclonal antibody prevents the cancer cells growing anymore. And as they're designed to only attach to the cancer cells, they should only affect the cancer cells and not healthy body cells. Indirect monoclonal antibody therapy can also be used, and this is when you would attach a drug to the monoclonal antibody. So in this instance, you've still created the antibody to be complementary in shape to the antigens on the cancer cells, but the cancer drugs are then delivered directly to the cancer cell, and it kills the cancer cell rather than lots of healthy body cells. So this should reduce the harmful side effects that affect traditional chemotherapy and radiotherapy can produce. And this is known as a bullet drug. It's going straight to where it's needed. So if you have a look at medical diagnosis, monoclonal antibodies can be used to test for things like pregnancy, influenza virus, hepatitis, chlamydia, prostate cancer, but also it's not on the spec because this happened after the spec was created, but COVID-19, all of those lateral flow tests that we used to have to do all the time use monoclonal antibodies. And this works via an ELISA test. So an ELISA test, the enzyme linked immunoabsorbent assay, it uses two antibodies. We can see here in the pregnancy test, we've got first of all a mobile antibody, and then we have a immobilized antibody. And we actually have a third one here. We've got an immobilized one as well. And in this instance, what happens is you would dip your pregnancy test into the urine that you are testing to see if someone is pregnant or not. Because when you are pregnant, you create this hormone HCG and it can be secreted in your urine. So this is absorbent paper. That means the urine with any hormones dissolved within it are going to move up through that absorbent paper. The hormone is complementary to the mobile antibody, which also has a coloured dye attached to it. So that will then move up the pregnancy test. And at point C, you have immobilised, meaning it can't move, antibody that is also complementary in shape to the hormone. So that will then cause the first antibody that has the hormone attached to it to attach to the immobilised one. And it also has that coloured dye on it. And that's why at that exact position you get a coloured line. Any antibody that didn't bind to the hormone will move past section C, but we do have antibodies also immobilised at point D that are complementary in shape to the end of that antigen. So those will bind and you get a second line. And in this way, whether you have that hormone or not, you should always get the line at D, and that is to help prevent false negatives, because it shows us that you don't have a coloured line here if you don't, but the antibodies are definitely able to move, so the test is working. Whereas if you have a line here, that means you are pregnant because you do have that hormone present. And this line here confirms that the test was working, so antibodies did move through the test. Now you can also have different versions of this. So we've got one here where we would add the test sample from a patient to the base of a beaker or the base of a well, and would then wash to remove any unbound test sample. Then you'd add an antibody complementary in shape to the antigen you are testing for, and that will bind if it's present. Again, wash to remove any that didn't bind. We then add a second antibody that is complementary in shape to the first one, so that's only going to bind if the antigen was present and therefore the first one bound. Again, wash to remove any that's unbound. Then you'd fill the wells with a liquid which is a substrate for this enzyme. And when those react together, you get a colour change. So the presence of the colour indicates that you do have that antigen present, and the intensity of the colour indicates the quantity present. Now, there are some ethical issues around monoclonal antibodies because creating monoclonal antibodies requires animals such as mice to produce those antibodies in tumour cells, which leads to ethical debates as to whether this use of animals is justified to enable better treatment of cancers in humans and detect disease. Because in order to do this, there will be suffering to the animal, and then at the end they would be killed because they wouldn't be needed. Topic 3 for AQA A-level biology, and we start with 3.3.1 surface area to volume ratio. Exchange surfaces in organisms, so where you might have diffusion occurring across, for example, have many similar adaptations to make that transport across the surface more efficient. And surface area to volume ratio is one of those adaptations. And the relationship between the size of an organism or the structure, so like the shape of a cell or the organism, and its surface area to volume ratio plays a significant role in the types of adaptations an organism will have. And we're going to see this repeatedly throughout topic 3. So the concept of surface area to volume ratio is if you were to work out the surface area of the object, and in the case of a cube, that would be the area of one side of the cube, so the length times the width, and then we times that by 6 because there's 6 sides to the cube. So our surface area for this cube is 6 centimeters squared. The volume would be the height times the length times the width, and 1 times 1 times 1 is 1 centimeters cubed. The surface area to volume ratio means divide the surface area by the volume, and that gives us 6, or a 6:1 ratio, but we can just present that as 6. If we then were to calculate that for these larger cubes, we can see this one, the surface area to volume ratio would be 3, and this one would be 2. And this is demonstrating a key pattern. The larger the object or the organism, so the bigger the size, the smaller its surface area is compared to its volume. And that means that small organisms, for example an amoeba, has a very large surface area compared to its volume. And this means that there is a big enough surface for exchange to happen across to compare to its volume. But also there's a smaller distance from the outside to the very middle of the organism. So you have a short diffusion distance as well. Now, as a result of this, very small organisms don't have special exchange systems that they've evolved. For example, a lung system or respiratory system, we should call that. The lungs is the organ. And therefore, they can just simply exchange substances across their surface. So as an organism increases in size, their surface area to volume ratio gets smaller. So that means larger organisms have a relatively small surface area compared to its volume. So they don't have a very big surface area available for exchange of substances like oxygen or heat compared to their volume. Whereas in the smaller organisms, as we said, they have a large surface area to volume ratio, and that enables efficient exchange directly across their body, so they don't have to have any specialised exchange or transport systems. Now, smaller mammals have a high surface area to volume ratio, and that means that they lose heat rapidly across their surface. So to replace that heat that they lose, they have to have a high rate of metabolism, and therefore they will respire rapidly. And this high metabolic rate provides the energy needed to provide more heat and maintain a stable body temperature, especially if they're in colder environments. Larger animals, because they have a smaller surface area to volume ratio, they will lose heat more slowly due to that. And their metabolic rate can therefore be relatively lower in comparison to the smaller mammals. So larger organisms then will have adaptations to increase the surface area to volume ratio at these exchange surfaces. So for example, villi and microvilli, which we'll see come up in the digestion topic in this video. Alveoli and bronchioles, spiracles and tracheoles, and gill filaments and lamellae, and also the thin wide leaves are all linked to gas exchange. This one here is in mammals, this one is in terrestrial insects, fish and plants, and this is all going to come up in this video. And then also many capillaries. So having a capillary network is going to be relevant at exchange systems for gas exchange, but also for the absorption of digested food. So these concepts will come up over and over again in this video. 3.3.2 is gas exchange, and some key terms to be familiar with this topic are breathing. This is the movement of air into and out of the lungs. Ventilation is the scientific term for breathing. Respiration doesn't really come up in this topic too much, but I just bring it up because that's commonly mistaken for breathing at GCSE. So let's just clarify. Breathing and respiration are not the same thing. Breathing is air moving in and out. Respiration is the chemical reaction that releases energy in the form of ATP. And then gaseous exchange, that is diffusion of oxygen from the air in the alveoli into the blood, and of carbon dioxide from the blood into the air in the alveoli. Now, gas exchange just means the exchanging of gases across a surface, and that will be by diffusion. So gas exchange surfaces all have the same three features that they'll have adaptations for. Large surface area to volume ratio, short diffusion distance, maintaining a concentration gradient. And when we have a look at the gas exchange system in mammals, for example, humans, in terrestrial insects, in fish, and in leaves, we'll look at what do they have as adaptations to meet these needs. And that's the common marking points that come up for all three. And it links to this equation here, which you don't have to know, but it just helps to explain it. The rate of diffusion is proportional to how big the surface area is, how steep the concentration gradient is, divided by the length of the diffusion pathway. So if we have a look at the human gas exchange system, these are the 5 structures that you need to be familiar with. So the alveoli, which are the air sacs. We then have the bronchioles that attach to those, and they're branching. You then have the bronchi, which is the 2 splitting tubes from the trachea, which is your windpipe. And the lungs, you have a left and a right lung. Ventilation is how we get air in and out of the lungs. And you need to know about the role and what happens to the diaphragm, the rib cage, and the intercostal muscles. So ventilation is inhaling and exhaling, and it is controlled by these muscles. And when it comes to the intercostal muscles, you have to be specific, talking about the external or internal. It's normally only the external that they refer to on the mark scheme. So let's have a look at these two. For inspiration, which is inhaling, the external intercostal muscles contract, and that pulls the rib cage up and out. That would be 1 mark. I've not seen on the mark scheme anything linked to the internal intercostal muscles, but just so you're aware, they'll be relaxed because they're antagonistic muscles, which means if one is contracting, the other does the opposite, so relax. The diaphragm is a muscle, so it contracts, and that then pulls it to move down and flatten. That would be a mark on the mark scheme. This then causes the air pressure in the lungs, or you could say the thoracic cavity, to decrease. And the reason the air pressure is decreased is because the lung volume has increased, or again, you could call that the thoracic cavity. Because that rib cage has moved up and out, the diaphragm has moved down, you now have a bigger space, so a larger volume, but it contains the same number of air particles. So that means that the pressure decreases. And for that reason, air moves in. So your key marking points would be this row here about the external intercostal muscles, this row about the diaphragm, and then these two combined. So that would be a 3-mark answer. Expiration or exhaling, exact opposite. So the external intercostal muscles relax, and that causes the rib cage to move down and in. The internal intercostal muscles, again, it doesn't really come up in the mark scheme that you'd have to say that they contract. So the pull the rib down and in, that would link to this mark here. The diaphragm relaxes, and that causes it to dome and move upwards. And because of that, you've now decreased the volume in the lungs or the thoracic cavity, and therefore that increases the pressure in the thoracic cavity. And because you've got a higher pressure, that is moving air out. Now, there is a maths equation linked to this, pulmonary ventilation, and that is the total volume of air that is moved into the lungs during 1 minute. So decimeter cubed is our unit for volume. Per minute is how we get it as a rate. So you'd need to do the tidal volume times ventilation rate. And the tidal volume is the volume of air inhaled or exhaled in one breath or breathing cycle. Ventilation rate is the number of breaths per minute. So if we have a look at some adaptations then to increase the rate of gas exchange or diffusion of the gases, once the gases are in the alveoli, so because of that ventilation, the gas exchange occurs between the epithelium and the blood. Alveoli are the tiny air sacs at the end of the bronchioles, and in each of your lungs you have about 300 million, and that is how we get the large surface area. One alveoli does not provide a large surface area, it's the fact that there are so many in each lung. That's adaptation number 1. The short diffusion distance, that is because the alveoli epithelium is just a single layer of cells. That's a very thin layer to minimise the diffusion distance. It's the same thing with the capillaries as well. It's just one layer of cells making up that epithelium. So that is our short diffusion distance. The maintaining the concentration gradient is nearly always the same adaptation, surrounded by a network of capillaries. Because what that means is, as the gas diffuses into the blood, it's then carried away and replaced by blood that is low in that gas, in this case oxygen. So those are our key adaptations for gas exchange in the human lungs. We then move on to gas exchange in fish, and fish get their oxygen from the water. The challenge they face though is there's about 30 times less oxygen dissolved in water than there is present in air. So they have to have special adaptations to really ensure that they maintain that concentration gradient even when there's so much less oxygen dissolved in water. And we'll come to that when we get to the countercurrent flow. So fish gill anatomy first then. So the gills are found on both sides of the head, and they're found as 4 layers of gills. The gills are made up of these stacks of gill filaments. So this long structure sticking out is a gill filament. Each gill filament, so we're zooming in on one gill filament here, has lamellae at right angles going all the way along it. And the lamellae is the site of gas exchange. So because you have gills on both sides of the head, you then have lots of gill filaments, and every gill filament is covered in lots of gill lamellae. That is what provides the large surface area for gas exchange. And when the fish opens its mouth, water rushes in and it goes through. There's a hole in the side of the head, which you can see here. So that causes the water to move in, goes over the gills and comes out the other side. And as the water flows over the gills, the gas exchange will happen at the gill lamellae. So other adaptations then. We've talked about how they have a large surface area to volume ratio. Next then is how is the short diffusion distance maintained or provided? And that is, first of all, because the gill lamellae are very thin, but also within every gill lamellae there's a capillary network, and the gases are diffusing from the water into the blood in the capillaries, and that's a really short diffusion distance. Finally then, it's how do they maintain that concentration and diffusion gradient across the entire lamellae, even though there's a low amount of oxygen in water. And that's when we get to the countercurrent flow mechanism. And the countercurrent flow mechanism means that the blood and the water flow in opposite directions. So this is when water flows over the gills in the opposite direction to the flow of the blood in the capillaries. That would be one of your key marking points for what is the countercurrent flow mechanism and how is it advantageous. That means that the equilibrium of oxygen is never reached, because as the blood flows in one direction and the water flows in the other, that means you should always have blood which has a lower concentration of oxygen reaching water which has a higher concentration of oxygen. So equilibrium is not reached. And your key marking point, your third mark, would be this ensures that a diffusion gradient is maintained across the entire length of the lamellae. Because if it was concurrent flow, that should be con, not co, that would mean that the blood and water flow in the same direction, and by the time you're about halfway or 3/4 of the way across the lamellae, you would reach equilibrium, so there'd be no gas exchange or diffusion in the end section. And that's why the key mark is across the entire length of the gill lamellae. Next, then we look at terrestrial insects. They have an exoskeleton made of hard fibrous material, and that is an advantage because it's protection and it prevents water loss, but it reduces the space that they could do gas exchange across. So they don't have lungs, and instead they have a tracheal system. And the tracheal system is made up of a trachea, tracheoles and spiracles. Whenever you're describing this in an exam question, there's normally one mark just for stating those three structures. So always start by stating those. The spiracles are these round valve-like openings that run along the length of the abdomen, and they can open and close like stomata in plants. And that is where the gases can enter and leave. The trachea is a network of internal tubes. So once the gases enter through the spiracles, they then go through the trachea, and they do have rings within them to keep them open the whole time. And then the trachea branch into tracheoles, and these are much, much smaller tubes that are going to reach the abdomen of the insect. And they extend throughout all of the tissue in the insect to ensure that oxygen is delivered to all respiring cells. So those are the three key structures. Now, in terms of the adaptations, we need to think about large surface area, short diffusion distance, maintaining the concentration gradient. So first of all, tracheoles, which are the branching structure where gas exchange actually happens, have very thin walls. So that provides a short diffusion distance. Tracheoles are also highly branched, which provides a short diffusion distance because they're reaching so many cells that there's never going to be a big distance between the tracheoles and these cells. And also because the tracheoles are highly branched, that provides a large surface area for gas exchange. So these are our adaptations for short diffusion distance and large surface area. Linking to the maintaining of a concentration gradient, at rest, so when the insect isn't flying, diffusion is the main method of gas exchange happening at those tracheoles, and when the cells are respiring, they're using up oxygen and producing carbon dioxide. And that is enough to create a steep concentration gradient from the atmosphere compared to the tracheoles for the insect. So that is how the concentration gradient is maintained, is through respiration. And that's when they're at rest. During periods of high activity, so for example when they're flying, there will be mass transport of the gases occurring. So what happens in this case is the insect's abdomen will contract and relax, which actively moves air in and out of the tracheal system, and that helps to maintain the diffusion gradient. Finally, when the insect is very active, so they might be flying, they will end up respiring anaerobically. And lactate or lactic acid is produced during anaerobic respiration. And when that dissolves in the cells, it's going to lower the water potential of those muscle cells. And this causes water, which is naturally present in the very tips of the tracheoles, to move into the cells from the tracheoles by osmosis. And because water has now left the tracheoles and gone into the cells, there is now an empty space to be filled with air in the tracheoles. And that is going to cause gases to move more quickly into the tracheoles. Now, when it comes to reducing water loss, in the spec, it says you need to know how terrestrial insects reduce water loss and plants, and you need to know comparisons between the two. So let's have a look at terrestrial insects first of all. Insects have that waterproof exoskeleton, and that prevents evaporation of water across their surface. The spiracles that they have can open and close, and when they close, that prevents water from evaporating out. So that reduces water loss. And the spiracles also have tiny, tiny microscopic hairs around them as well, and that will trap any humid air, reducing the water potential gradient and therefore reducing evaporation. We'll come to the examples of the plants once we've gone through gas exchange in plants. So the structure, or the cross-section of a leaf, we have at the very top the waxy cuticle, the upper epidermis, the palisade mesophyll, which is the site of photosynthesis, spongy mesophyll, that's where we have lots of air spaces. We then have the lower epidermis, and within the lower epidermis there are guard cells which create these pores known as stomata. This bit here is the vascular bundle, which is made up of the xylem and the phloem. And we can see those structures here under the microscope as well. So gas exchange in plants then, the palisade mesophyll, as we just said, that is the site of photosynthesis. So that is where lots of oxygen is going to be produced and carbon dioxide is going to be used up. That is what is creating the concentration gradient. So because we have that concentration gradient, oxygen will then travel through the air in those spaces in the spongy mesophyll and diffuse out of the stomata created by the guard cells, assuming that oxygen is not being used in respiration. Carbon dioxide, because that's being used up, we also have a concentration gradient compared to inside the leaf and the outside, so carbon dioxide will diffuse in through the stomata. So that is how the concentration gradient is maintained. To reduce water loss, the stomata will close when it is not light, so at night, because they won't be photosynthesizing, so they don't need carbon dioxide. The short diffusion distance is the fact that leaves are so thin, so it's a very short distance for the gases to diffuse from the outside in the atmosphere through the stomata to the palisade mesophyll. And the large surface area, leaves are very broad, so you're going to have lots of these cells and also lots of stomata, so there's a large surface area for that gas exchange as well. So the gas exchange at the stomata, oxygen diffuses out, carbon dioxide diffuses in, and we've just said to reduce the water loss, stomata close at night. Now, the other adaptations, in particular in xerophytic plants, which are plants that are adapted to survive in really hostile abiotic factors where there's very little water, the structural adaptations that they have are, first of all, a lot of the time the leaves curl up, and that means all of the stomata are facing inwards, and any water that does evaporate gets trapped, and it creates a very humid atmosphere to reduce the water potential gradient, so less water evaporates out. You also have these hair-like structures, which again trap humid air to increase the humidity. Sunken stomatas, again, that increases the local humidity. And you have a thicker waxy cuticle as well to help reduce evaporation too. You do have a longer root network. Now that's not going to reduce water loss, but that means you can reach more water to more readily replace the water that might be lost. Next then, 3.3.3, digestion and absorption. So during digestion, the definition of digestion is large biological molecules are hydrolyzed into smaller molecules that can be absorbed across cell membranes. And in mammals, we will learn about how carbohydrates, lipids, and proteins are digested. Those are the three biological molecules that you need to know. So first of all, the carbohydrates, they require more than one enzyme to hydrolyze them into the monosaccharides. It's amylases and membrane-bound disaccharidases. And the amylase is produced by both the pancreas and the salivary glands. So that's why digestion begins in the mouth, because the salivary glands will release amylase, and then in the mouth, any starch that is there will start to be digested. That will then be swallowed, goes into the stomach. The stomach acid will denature that amylase, and that is why the pancreas then releases more amylase into the very top part of the small intestines known as the duodenum, and you do have to say the duodenum to get the mark. And because we've now got more amylase being released there, any remaining starch can continue to be digested. Then as that food travels into the next part of the small intestines called the ileum, most of that starch should have now been digested into disaccharides, and embedded within the membrane of the epithelium of the ileum, you have membrane-bound disaccharidases, meaning enzymes embedded within that membrane that can digest the disaccharides into monosaccharides. So that could be the enzyme sucrase, digesting sucrose, or lactase, digesting lactose, or maltase, digesting maltose, all of which will hydrolyze the disaccharides into the monosaccharides, which are then small enough to be absorbed from the ileum into the bloodstream. So here are the three word equations from biological molecules, but just as a reminder here, because this could come up on paper 1, so that means even though this is paper 1 theory, it could come up linked to digestion, knowing what the 3 disaccharides are digested into or hydrolyzed into by those membrane-bound disaccaridases. Next then, proteins, and this starts in the stomach, it continues in the duodenum, and then it's fully digested in the ileum of the small intestines. And there's 3 enzymes involved in hydrolyzing these large polymers into the monomer amino acid. We have the endopeptidases. These hydrolyze the peptide bonds between amino acids in the middle of the polypeptide chain. That means that you're going to be breaking or hydrolyzing one long polypeptide chain into multiple shorter chains. And this increases the surface area for the next enzyme to work with. And that is the exopeptidases. And these hydrolyze peptide bonds between amino acids at the end of the chain. So by hydrolyzing the chain into many smaller ones, that's what the endopeptidases do, that increases the number of exposed ends for the exopeptidases to work on. So those two work together, doing the same thing, hydrolyzing peptide bonds, just in different locations to speed up protein digestion. Now, if there are any dipeptides remaining, meaning 2 amino acids still joined together, by the time that protein or digested protein then reaches the ileum, it's the membrane-bound dipeptidases, which are enzymes that can hydrolyze the peptide bond between 2 amino acids. Those enzymes will work to hydrolyze a dipeptide into amino acids, and the amino acids are small enough to be absorbed. So in terms of these proteins, you would have in the stomach the endo and exopeptidases. The pancreas will release more of those into the duodenum, and then in the ileum you have your membrane-bound dipeptidases. Next then is the lipids, and these are digested by only one enzyme, lipase, but also by the action of a chemical called bile salts. So lipase is produced by the pancreas and it releases it into the duodenum. And lipase can hydrolyze the ester bonds in the triglycerides. And that's the bond between the glycerol molecule and the fatty acids. So by hydrolyzing those ester bonds, you are hydrolyzing and releasing those fatty acids, and you do get monoglycerides as well. Bile salts are not enzymes, but they are produced by the liver. That will then be released into the gallbladder, and then the gallbladder releases it into the duodenum. And what bile salts do is emulsify these large lipid droplets into smaller droplets to increase the surface area for lipase to work on. They're also important in the formation of micelles. That comes up in lipid absorption rather than digestion. So this can be split down into the concept of physical and chemical digestion. The physical digestion is this emulsification. So the bile salts produced by the liver emulsify the lipids, meaning splitting it up into smaller droplets, but no chemical bonds are broken. So it's not chemical digestion, it's just splitting it up into smaller droplets so that there's faster hydrolysis by lipase. Chemical digestion is when bonds are broken, and lipase hydrolyzes the ester bonds so that we get glycerol, fatty acids and monoglycerides. A monoglyceride is the glycerol molecule with one fatty acid still attached. Next then we move on to absorption, and in mammals the products of digestion are absorbed across the cells lining the ileum, which is within the small intestines. So adaptations to increase absorption, and this is going back to this concept of this is an exchange surface, so we need to look for adaptations that increase the surface area, reduce the diffusion distance, and maintain the concentration gradient. So the ileum wall is covered in villi, which are these foldings, which have thin walls surrounded by a network of capillaries, and also these epithelial cells have even smaller microvilli, which is the folding of the cell membrane. And it's those features that maximize absorption. So the increased surface area is the villi and the microvilli, because you've got that highly folded surface. Decreasing that diffusion distance is because there's thin walls, but also the capillary network branches all the way up into the villi. So it's a short distance to diffuse across to reach the capillary network. Maintaining the concentration gradient is also because of that capillary network, because any digested food molecules that do get absorbed are transported away in the blood in the capillaries. Now I've also got a table of adaptations listed here that I repeatedly have seen come up in the mark scheme. So you can screenshot this, turn it into a flashcard. These are what come up in long answer questions, short answer questions for adaptations to maximize absorption of digested products. So learn that. So now we've had a look at the adaptations for absorption, we need to know how the products of lipid digestion are absorbed, and also how the products of carbohydrate and protein digestion are absorbed. Starting with lipid absorption. And now we get to find out more about what are micelles. So micelles are water-soluble vesicles. And these vesicles contain fatty acids, glycerol and monoglycerides. Those are the products of lipid digestion. So your triglyceride, after it's been digested, that's what we have. And it's also bile salts combined with those. And it's the bile salts that make these products, these fatty acids, water soluble. So that's what a micelle is, but a common exam question is, how do micelles help with lipid absorption? And they do it in these ways. So we've just said that the micelle is fatty acids, monoglycerides, bile salts, and those bile salts help to make the fatty acids soluble in water. Now that is an advantage for lipid absorption because it allows the micelles to move the fatty acids and monoglycerides from wherever in the lumen they were digested towards the cell surface membrane of the epithelial cell. Because the contents of the lumen, which is like the hollow space, the hollow tube of your small intestines, that is containing lots of watery substances from digestion. And fatty acids are not soluble in water. So we need them to be soluble so they can move easily towards the epithelial cell surface membrane. So that's why being water soluble is an advantage. So the second mark links to that. The micelles carry and then release the fatty acids and the monoglycerides to the epithelial lining of the ileum. So that then means the fatty acids are all released at this point. It's actually often another marking point from here that it helps to maintain the concentration gradient of the fatty acids near the epithelial cell. And the fatty acids, monoglycerides and glycerol, those are then absorbed by simple diffusion. The micelle does not enter, the micelle releases those at the cell lining. So those will then enter by simple diffusion because The cell membrane is made up of phospholipids, and only non-polar molecules can diffuse through those, and fatty acids are non-polar. So inside of the cell, once it's been absorbed in the cell, it will then be reformed into triglycerides, and that happens in the Golgi body. So we get them packaged into these triglycerides, into vesicles, and those vesicles containing the packaged triglycerides will then move towards the cell surface membrane on the other side, fuses with the membrane, it then releases that triglyceride, which is picked up by a lacteal rather than the blood in the capillaries, and that is the end point of the absorption. Then we need to look at how the products of carbohydrate digestion and protein digestion are absorbed. So products of carbohydrate digestion are monosaccharides, for example glucose. Products of protein digestion are the amino acids. And both of these are absorbed into the blood in the same way, and it is co-transport, which you learn in topic 2. So for co-transport of glucose, and it'd be exactly the same for amino acids, first of all, we have sodium ions from the epithelial cell actively transported into the blood. This creates a concentration gradient of sodium ions from the ileum into the epithelial cell. And therefore the sodium ions can enter the epithelial cell down their concentration gradient, but they are an ion, so they have to go through a protein. And the protein that they can transport through is a co-transporter protein, which will bind sodium ions and glucose or amino acids. And that is then how the glucose and if it was amino acids enter the epithelial cell. Once inside of the epithelial cell, we already have a concentration gradient then. There'll be lots of glucose in the cell compared to in the capillary. And that means then that glucose can move down its concentration gradient into the blood and the capillary by facilitated diffusion through a transport protein. And it has to be facilitated because glucose is too large to move by simple diffusion through the phospholipid bilayer. Next then, 3.3.4 mass transport. Starting with hemoglobin, and these are groups of proteins found in different organisms. And hemoglobin is a protein with a quaternary structure. Quaternary structure from topic 1 means a protein with more than one polypeptide chain. And hemoglobin has 4 polypeptide chains, each of which has a heme group attached, which is where you have the iron-ion. And it's this heme group and the iron-ion where oxygen binds. So one hemoglobin molecule can bind 4 oxygen molecules, and then it transports the oxygen to respiring cells. Myoglobin sometimes comes up in application questions. So just to give you an awareness, this is another protein that can transport oxygen. It's not hemoglobin, but it's like hemoglobin in that it can transport oxygen, but it can only transport 1 oxygen molecule at a time. And some animals have a lot of this, and we have it in our muscle tissues. So oxyhemoglobin dissociation curves. Oxygen is going to be loaded in areas in the body with a high partial pressure of oxygen. So for example, the alveoli in the lungs, and it will be unloaded in regions of low partial pressure. So where oxygen's been used up for respiration. So on our oxyhemoglobin dissociation curve, this axis is showing us the percentage saturation of oxygen on the hemoglobin at different partial pressures of oxygen. And that basically means the amount of oxygen present. Partial pressure is a way to consider concentration. So in areas where there's a low partial pressure, meaning not a lot of oxygen present, that would be respiring tissues because oxygen's constantly used up in aerobic respiration. And at areas with a higher partial pressure, for example, the alveoli in the lungs, that is because as you inhale, you're getting new air, which will have lots of oxygen within it. So this oxygen dissociation curve, or oxyhemoglobin dissociation curve, it shows us that at low partial pressures, hemoglobin is not as saturated as it is at high partial pressures. And if it's not as saturated, that must mean it's either unloading oxygen or not loading oxygen as much. And we'd say at that point, that would mean the hemoglobin has a low affinity for oxygen. Affinity being the attraction and ability to bind oxygen. Whereas at higher partial pressures, because the haemoglobin is nearly fully saturated with oxygen, that must mean that haemoglobin has a very high affinity for oxygen. It's loading lots of oxygen onto it. Cooperative binding is this concept of when you have different numbers of oxygen bound to the haemoglobin, it affects the ability of subsequent oxygen molecules to bind. So haemoglobin can associate with, which means load, 4 oxygen molecules because of those 4 heme groups. And as each molecule binds, it slightly changes the shape of haemoglobin, exposing the further binding sites, making it easier for each subsequent oxygen to bind. So if we think of this as, we've got 0, let's say that's the first one binding, second oxygen binding, third and fourth. We can see that we've got not a particularly steep gradient compared to the point of 2 and 3, showing that it must be hardest for the first oxygen to bind. And that's because at this point the binding sites are slightly covered. Once that first oxygen binds, it changes the shape and exposes the binding sites. So we can see we've now got a steeper gradient, and that is representing that it's easier for the second and the third oxygen to bind. However, when we get to the fourth oxygen, we can see the gradient is less steep again and then it plateaus. And that's because if 3 of the 4 binding sites are already saturated, you've got a 1 in 4 chance now of the oxygen binding with an empty binding site. And that's why the ability to bind the fourth one is also more unlikely or challenging. So this all shows us that in areas with a high partial pressure of oxygen, as we said, there'll be a higher affinity. In areas with a lower partial pressure, there'll be a lower affinity. The Bohr effect is looking at the effect of carbon dioxide dissolved in the blood on haemoglobin's affinity. And when carbon dioxide dissolves in the blood, and by the way, this carbon dioxide is coming from aerobic respiration. So when you're respiring lots aerobically, you will have a lot of carbon dioxide dissolving in your blood, forming carbonic acid. Carbonic acid decreases the pH of the blood, and that decrease in pH slightly changes the tertiary structure of the polypeptide chains in haemoglobin. And this is why the affinity for oxygen decreases in respiring tissues. That change in shape is going to decrease the affinity. So haemoglobin ends up loading less oxygen and unloading more. And that is an advantage because if haemoglobin is going to be unloading oxygen more readily at respiring tissues, that means it's delivering the oxygen to the site of aerobic respiration where it's needed, so aerobic respiration can continue. And the way we can see this on the graph is, as the pH becomes more acidic, we can see the curve shifts to the right each time. And the best way to consider this in an exam is, at any of the partial pressures, use your ruler, which I don't have right now, and draw a vertical line and read off what the partial pressure is. Sorry, the saturation, not the partial pressure. So read off what the saturation is for that partial pressure. And let's say this one is maybe 25, 39, and that might be 56. That's very inaccurate readings off. But the point is, even at the same partial pressure, the most acidic is 25% saturated. The least acidic is 56% saturated. So what this shows us is the haemoglobin in the more acidic conditions must have a lower affinity, indicated by the fact it's less saturated, so it's more readily unloading oxygen rather than loading. So if a curve shifts to the right, that is what it's indicating, and this is how I demonstrate it in an exam. Read it off and show that even at the same partial pressure, the saturation is lower, showing that it must have a lower affinity for oxygen. Within the spec, it does say you can get applications linked to different haemoglobins for animals. So it says you need to know that animals are adapted to their environment by possessing different types of haemoglobin with different oxygen transport properties, which basically means based on their environment, they've evolved to have haemoglobin with either a higher affinity for oxygen if they live in an environment which has a low partial pressure of oxygen, Or they might have evolved to have hemoglobin with a lower affinity for oxygen if they need to readily unload it for constant aerobic respiration at higher rates. So things like small organisms that need to maintain their body temperature and they lose more heat across their surface. You might also have questions linked to fetal hemoglobin and myoglobin as well. Myoglobin has a very high affinity for oxygen, even at very, very low partial pressures, and in that way it can act as an oxygen store. Linked to the fetal hemoglobin, this is showing us in blue the curve for fetal hemoglobin and green, the adult hemoglobin, and that would be the mother, for example. And a fetus doesn't breathe. It gets all of its oxygen from the placenta as the blood then goes from the placenta, passes across the umbilical cord and into them. And that means that it's really important that a fetus has hemoglobin with a higher affinity for oxygen than the mother's hemoglobin's affinity for oxygen. Because that then will show us, again, I'd draw a line. Let's say we were at 25 partial pressure. We can see that we're maybe 45% saturated, whereas here we're 70%. So that would mean at this partial pressure, some of the oxygen is being, almost half of the oxygen, in fact over half of the oxygen has been unloaded from the mother's hemoglobin. And that can then be picked up by the fetus's hemoglobin because they are still at a high saturation, indicating a higher affinity. So at these lower partial pressures, the adult, the mother hemoglobin, will be unloading oxygen, but the fetus's hemoglobin, which has a higher affinity even at low partial pressures, will be able to load up the oxygen still. And in that way, a fetus gets oxygen from the mother. So we move on to the circulatory system, and mammals have a closed double circulatory system. Closed meaning the blood remains within the blood vessels and the heart, whereas the double circulatory system means the blood passes through the heart twice in each circuit. So one circuit goes to the lungs, one circuit goes to the rest of the body. And mammals require a double circulatory system to help manage the pressure of the blood flow. So the lungs are directly behind the heart. So they're not a very far distance at all. So you don't need a high blood pressure to make sure that the blood reaches the lungs. And in fact, we don't want a high blood pressure because that could damage the capillaries in the lungs if the blood was pumping through at that high pressure. And also it's an advantage to have a lower pressure because that reduces the speed at which the blood flows, which means there's more time for gas exchange in the alveoli. In contrast, we do want a high blood pressure for the blood flowing in the circuit from the heart to the rest of the body because we need it to ensure it can reach all of the respiring cells in the body. You need to know some key blood vessels. It's not all of the ones in the body that we can see here, but it's the ones in the red boxes. So you need to know the coronary arteries, which we can't see here, but these are the blood vessels that are on the outside of the cardiac muscle on the heart, and it supplies the cardiac muscle itself with oxygen for respiration. And then you need to know the blood vessels that are connected to the heart, the lungs, and the kidney. So we'll start at the top of the lungs. Any blood vessel that is connected will have pulmonary if it's for the lungs. And artery means it's blood coming away from the heart. Vein, if you think in, it's going back into the heart or towards the heart. So pulmonary artery is blood coming away from the heart to the lungs. And pulmonary vein is the blood vessel that is taking blood from the lungs back into the heart. Then if we move on to the heart, we've actually covered two already because the two that are connected to the lungs are also two connected to the heart. So then if we have a look at the blood vessels carrying blood away from and into on this side, the aorta is the artery carrying blood away from the heart to the rest of the body. The vena cava is the vein carrying blood from the body back into the heart. Vena cava meaning body vein. And lastly, the kidneys. Any of the blood vessels that are attached to the kidneys have renal in front of them. So renal vein means it's the blood vessel from the kidneys taking blood back into or towards the heart. And renal artery means it's blood coming away from the heart to the kidneys. So those major blood vessels are then connected within the circulatory system via arteries, arterioles, capillaries, also venules, but you don't need to know that for AQA, and veins. And that takes us on to these blood vessels. So arteries in general, away, they carry blood away from the heart towards the arterioles. Arterioles are smaller than arteries, they're branching and they connect to the capillaries. You don't need to know about the venules, but you then have the capillary network, and then you learn about the veins, which carry blood towards the heart. And I always say, like, vein, it's got in it, it's going back in or towards the heart, away. So we can see this here just from another angle, and it's showing us the inside of the walls, which are the adaptations that you need to know for the blood vessels. Now, the aorta is the largest artery in the body. It carries blood at the very highest pressure in the body. And it carries it at high pressure because it's carrying it directly from the heart, where that left ventricle would have just contracted, forcing the blood out at high pressure. So it contains a large amount of elastic tissue in its wall, which means that it can stretch when the ventricle contracts, and it can recoil when the blood is then flowing away, causing it to move back to its original shape. And this helps to smooth out blood flow and also maintain consistent pressure. It's got a very thick muscular wall as well, and that helps it to withstand high blood pressure and prevent it from bursting, whilst also the muscular wall can contract, and that helps to smooth the blood flow as well. Arteries in general, aorta is an artery, but it's the thickest or it's the biggest. Arteries all have thick muscular and elastic walls. That means they've got a narrow lumen for the blood to flow through, and it helps them to maintain the high blood pressure as they are directing the blood from the heart away to all of the cells of the body. Arterioles are going to be branching from the arteries and help to regulate blood flow into the capillaries. So they still have a relatively thick layer of muscle, and that can contract to narrow the lumen or vasoconstriction. That reduces the blood flowing into the capillaries. And when the muscle relaxes, the lumen widens, which is vasodilation, increasing the blood flow to the tissues. Lastly, in the veins, these carry blood back to the heart under low pressure. They have thinner walls. They still have muscle and elastic within the walls, but just a much thinner layer. And because it's a thinner wall, they have a wider lumen. And this is to reduce resistance of the flow. And that's because the blood is going to be flowing at a much lower pressure because it's now further away from the heart, which was the pump that increased the pressure in the first place. It also has valves which prevent the backflow of blood. So here's a little summary table of some of those features that you can screenshot and add to your notes. Capillaries then. These are the smallest of the blood vessels and adapted for efficient gas exchange or other exchange of substances between the blood and surrounding tissues. And some of the adaptations are, first of all, very narrow diameter, which means only one red blood cell can fit through at a time. That slows down the flow of blood, which is good because that means there's more time for exchange, for example, gas exchange. The walls are also only one cell thick, so it's consisting of a single layer of flattened endothelial cells, and that reduces the diffusion distance. Capillary beds are networks of capillaries that spread through tissues, and that provides a large surface area for exchange as well. And the extensive branching also means that all cells are going to be close to a capillary, and that is reducing the diffusion distance. So again, we're thinking here about large surface area and reducing diffusion distance. This leads us into tissue fluid. Tissue fluid is a fluid surrounding your tissues that contains water, glucose, amino acids, fatty acids, ions, oxygen that have all been forced out of the blood in the capillaries. So you need to know, how is tissue fluid formed, and then how does the water from the tissue fluid get reabsorbed? So in terms of the formation, the capillaries, we've said that it's only a single layer of cells making up that endothelium wall, that lining. And between every cell, there's going to be these microscopic gaps. So any molecule that's small enough to fit through those gaps can be forced out if there's a high enough pressure. And that's what we have at the arterial end. So this is where we'd have arteries, that branches into arterioles, and then that branches into the capillaries. And at the other end, the capillaries, then there's the venules and then the vein. At the arterial end, there is a high pressure. And that high pressure is originally generated from the contraction of that left ventricle in the heart. And then also the arterioles have got a wider diameter than the capillaries. So going from that larger diameter to a smaller diameter with the same volume of blood increases the pressure. And we call this high hydrostatic pressure. And because there's such high pressure, anything that's small enough to fit through those gaps between the cells and the capillaries is forced out, and that's known as ultrafiltration. Now, the key thing here to get the mark, you would have to name what gets forced out, not just say small molecules. So always name water, and then I would name at least 2 other things from this list. So water, glucose, amino acids are forced out. That is how tissue fluid is formed. So what is forced out then? Water, dissolved mineral ions, glucose, small proteins can, amino acids, fatty acids and oxygen. But anything that's too big to be forced out will remain in the blood in the capillaries. And that is primarily large proteins, also red blood cells and platelets, but the marking point is normally large proteins. And this is important because it helps to explain how the water from tissue fluid gets reabsorbed. So because those proteins remain in the blood in the capillaries, you end up with a much lower water potential at the venule end of your capillary compared to the water potential of the tissue fluid. And that is how water can then move from the tissue fluid back into the capillaries by osmosis. Now, in terms of mark scheme language, you must say it's water that is re-entering, not tissue fluid. So it's water that is entering by osmosis because of this decreased water potential. Now, within that water, there might be dissolved substances that are also reabsorbed, but that's not what you need to say. Because this is osmosis, that means eventually you will reach equilibrium in the water potential because water is moving in by osmosis. And when equilibrium is reached, no more water from that tissue fluid can be reabsorbed. But we need to reabsorb as much of it as possible, otherwise you get swellings in the body where you've got too much water outside of the blood vessels. And that's where the lymphatic system absorbs the rest. So then we move on to the heart, starting with the cardiac muscle that it is made from. So you get these thick muscular layers of cardiac muscle, and that has unique properties such as it's myogenic, which means it can contract and relax without any input from the nervous system or hormonal stimulation. So as long as it has a constant supply of glucose and oxygen in the blood in the coronary arteries, it can contract and relax. So it never fatigues as long as it does have that oxygen and glucose for aerobic respiration to make ATP so it can contract. So that's then our coronary arteries and the cardiac muscle. And this is just showing us the coronary arteries in more detail. So these branch off from the aorta, carrying oxygenated blood and also glucose that's needed for aerobic respiration. If one was to get blocked, this is now actually application, it's not on the spec anymore, but if you did have a blockage in your coronary artery, that means that all of these blood vessels would not have oxygenated blood flowing through them anymore. So all of that cardiac muscle would not get oxygen, glucose, wouldn't be able to respire, and it would stop contracting. And that is what a heart attack is, or myocardial infarction. It's when the cardiac muscle stops contracting. In terms of the structure of the heart, you have 4 chambers. So you have the 2 atria at the top, the 2 ventricles at the bottom. The atria have much thinner muscular walls, and that's because they don't need to contract with much force at all, because they're only moving the blood from the atria directly down into the ventricles. And they're elastic walls to stretch when the blood enters. Ventricles have much thicker cardiac muscle, and that's so that they can contract with more force because they're pumping the blood further. The right ventricle is pumping blood to the lungs are directly behind the heart. So it doesn't have to pump a very far distance. They don't need as thick a muscular wall compared to the left ventricle, which does have to contract with a large force to create a high blood pressure to pump the blood to all of the respiring cells in the body. So the left ventricle wall has the thickest cardiac muscle. Next is knowing the different blood vessels, which we actually touched on already because we've talked about the pulmonary arteries are carrying blood away from the heart to the lungs. Pulmonary veins carry blood from the lungs back into the heart. The aorta is the artery that carries blood from the left ventricle to the rest of the body. And the vena cava carries blood from the body back into the heart. Next is looking at the valves in the heart. So the semilunar valves are the ones between the ventricles and the arteries. So you have one in the aorta, which you can't actually see in this diagram, but it's just about here. And then we've got one here in the pulmonary artery, just there. The atrioventricular valves are found between the atria and the ventricles, and we have them both on the left and the right. The bicuspid is the name for the one on the left side, and the tricuspid is the name for the one on the right, but it's sufficient just to call them both atrioventricular for AQA. Now, valves will open when the pressure is higher behind them, forcing them open, and a valve will close when the pressure is higher in front of them. And in this way, they control the flow of blood so that it only goes in one direction, and it prevents the backflow of blood in veins, but also within the heart, making sure that the blood does go from the atria into the ventricles, out of the arteries. The septum, now this isn't technically on the spec, but just to make you aware, the septum is the part of the cardiac muscle that runs down the middle, separating the left and the right side, meaning the oxygenated blood from the deoxygenated blood, and that is going to help maintain concentration gradients for diffusion, because you're going to have far higher concentration of oxygen being pumped around, whereas if this all mixed together, it's going to decrease the concentration of oxygen in the blood. We then move on to the cardiac cycle, and there are three stages: diastole or diastole, atrial systole or systole, ventricular systole or systole. People pronounce it differently, so I'm going to keep switching between the two because I haven't decided how I'd pronounce it yet. So if we start with diastole, this is relaxation, and diastole is when both the atria and ventricle cardiac muscle are relaxed, and all of this pale green bit is our ventricular diastole, and all of this section is atrial diastole. But diastole means the cardiac muscles are relaxed. And this is when the blood is going to be entering from the body into the atria, when we've got both the atria and the ventricles in diastole. And it's going to enter through the vena cava and the pulmonary vein into the right and the left atria. The blood flowing into the atria is going to increase the pressure within the atria compared to the ventricles, and that causes the atrial ventricular valves to open, and so some blood will start to move into the ventricles at this point. Atrial systole, wherever you see systole, it means contract. Atrial systole is a very, very brief period. It's not a long stage, and this is when the muscles in the walls of the atria contract, increasing the pressure further, forcing all of the remaining blood that was within the atria into the ventricles. The ventricular muscle walls are relaxed at this point. You can see they're still in diastole. Then we have ventricular systole. So this is when the ventricles contract. And this happens after a short delay, meaning the atria contract, there's a short delay, and then the ventricles contract. And as they contract, that's going to increase the pressure in the ventricles to the point where the pressure is now higher in the ventricles than in the atria. And that is what causes the AV, or the atrioventricular valves, to close. And then when the pressure is even higher compared to the arteries, it causes the semilunar valves to open. and that is what causes the blood to be pushed out of the ventricles through the two arteries, either to the lungs or to the rest of the body through the aorta. Now, there is a math skill linked to this, the cardiac output, and that is the volume of blood which leaves one ventricle in 1 minute. And it can be calculated by doing the heart rate times the stroke volume. And heart rate is beats of the heart per minute. Stroke volume is the volume of blood that leaves the heart each beat. The final part is mass transport in plants. So we're going to look at transpiration first, the mass transport of water. And transpiration is the loss of water from the stomata by evaporation. And there are 4 key factors that can affect the rate of transpiration. Light is one of them, because the brighter it is, the more the stomata open. So there's a larger surface area for that evaporation or transpiration of water. Temperature, if you've got a higher temperature, there's more kinetic energy, so the water molecules will move faster. So again, more evaporation and transpiration. Humidity, if it's more humid, there'll be less transpiration. And that's because the more humid it is, the more water vapour in the air, and that will reduce the water potential gradient, so you get less water evaporating. The windier it is, that's going to blow away humid air containing that water vapour, and that can help to maintain the water potential gradient, so increasing that rate of transpiration. Then if we have a look at the movement of water up the xylem, water has to move from the roots all the way up a plant against gravity. And sometimes it's a small plant, sometimes it's a plant that might be several meters tall, moving water against gravity. And this is possible because of the cohesion tension theory. And that's what we're going to have a look at next. What cohesion is, what capillarity or adhesion is, and what we mean by the tension part in this theory. Now, this links back to module 1 with the properties of water. Water is a polar molecule, meaning it's got a slight negative charge on the oxygen atom, slight positive charge on the 2 hydrogen atoms. And for that reason, hydrogen bonds can form between the oxygen and hydrogen of different water molecules. That is cohesion, those hydrogen bonds between water molecules sticking the water together. And that means that water can move up the xylem as a continuous column of water. Now, you also get hydrogen bonds forming between the water molecules and the walls of the xylem, and that is adhesion, the sticking of those water molecules and then the entire water column to the walls of the xylem. And that can result in the xylem being drawn in more and more narrow, because we're going to look at how the water column gets pulled. And if you're pulling upwards on that column and the column is all attached and it's stuck to the wall, as you pull it up, it's like an elastic band. It's going to cause the xylem to become longer and narrower. And the reason that helps move water up against gravity is the narrower the tube, the more surface area you have for the water column to stick to, and that means it can actually move up the tube with more ease, which is just shown here with like different diameter straws. So the cohesion tension theory then. Water evaporates from the stomata. That is transpiration. That would be your first mark. Water evaporates from the stomata. As that water evaporates, it decreases the water potential in those surrounding cells, and that creates tension in the water column and on the xylem, and that draws and it pulls the water up. So that'd be your next mark. As that water is lost by transpiration, that creates tension on the water column and it pulls the water column up. The water molecules stick together because of cohesion. So that's your third mark. All of those water molecules form a continuous water column because they stick together because of the cohesion by the hydrogen bonds. And then at the same time, we do also have the column of water sticking to the walls of the xylem, adhesion, and that helps to move it up, and it also draws in the xylem narrower under that tension. So that is our cohesion tension theory. As I've read this through, this is a lot of text here. That's to give you the full understanding, but the bits that are in bold, those are your key marking points typically, and the ones that I pointed out as well. So then we have a look at the mass transport of sucrose in plants. And this is one of our products of photosynthesis. And all cells need this sucrose to then break down or hydrolyze into glucose for respiration. So this is going to be transported from the leaves, where the organic substances are made in photosynthesis, to all the respiring cells through the phloem. And the sugary solution, that sucrose solution, can move up or down through the phloem, whereas in the xylem, it only ever moves upwards. So the phloem tissue, there are two key cell types. We have the sieve tube elements and the companion cells. The sieve tube elements are here in the middle. They are living cells, but most of the organelles have disintegrated. So there's no nucleus and they don't have many other organelles. And that's why they need these cells next to them, called companion cells. And the companion cells will have lots of mitochondria in them so that they can do lots of aerobic respiration and provide the ATP needed for active transport in this process. So this links to the mass flow hypothesis, and this is looking at how the sucrose that's made in the leaves, which is our source, can be transported to the respiring cells, which is known as the sink. So we're going to go through this process of how it happens. There's a lot of text here, which I'm not going to talk through at this point. By all means, you can pause, write it down, but it's also going to come up in the subsequent slides. So I'm going to talk through it with those. This has just got a lot of the information in one place for you to screenshot if you want. So the source to sink explanation with this model helps to explain the mass flow hypothesis in terms of how the hydrostatic pressure causes the mass movement of sucrose solution. So in this tank, this model, we've got a tank of water. We've got one tube representing the phloem, another tube representing the xylem. And our source cell would be our photosynthesizing cell, where you'd have lots of sucrose being produced. And sucrose lowers the water potential of the cell and also of the phloem. And that means water is going to enter from the xylem into the phloem. At the sink cell, because lots of respiration is occurring, that's going to be using up sucrose, and that means you will now have a more positive water potential, or less negative. So water will actually move from the phloem in this region back into the xylem, and that will decrease the pressure. So at this end, we have an increase in the hydrostatic pressure as water moves in by osmosis. And at this end, we have a decrease in hydrostatic pressure because water leaves by osmosis. And as a result, because we've got high pressure and low pressure, that forces the liquid through the phloem from the source to the sink. So you also need to know, well, what is it that is creating this hydrostatic pressure and how does the sucrose move? So our source cell, we're looking at it slightly differently here now because it's actually in the plant rather than a model. The source cell is the leaf. That's where photosynthesis happens, sucrose is produced. Sucrose is actively transported from the source cell into the sieve tube element. So it has to go from the source to the companion cell to the sieve tube element. And that is done using ATP because it's active transport. And the ATP is provided by the companion cells. So this is your mark number 1 in translocation. Sucrose is actively transported using ATP from the companion cells. Now we've got the sucrose into the sieve tube element. That lowers the water potential of the sieve tube element, and therefore water from the xylem will enter the sieve tube element by osmosis. So here would be our mark 2. That increases the hydrostatic pressure. That would often be mark number 3, or these 2 points will be split between 2 and 3 marks. And because we've got a high hydrostatic pressure, that forces that sucrose solution to move en masse, so this is our mass transport, towards the sink cell. And that is because at the sink cell, sucrose is being used up in respiration, and therefore we're going to have sucrose from the phloem being transported into the sink cell, initially by facilitated diffusion, but when the concentration gradient is equal, you have equilibrium, you'll then have active transport of the sucrose from the sieve tube element into the sink cell. But either way, that means you have less sucrose here. That now means you have an increase in the water potential gradient, and water will move from the sieve tube element by osmosis back into the xylem. So it's that pressure gradient, the high hydrostatic pressure compared to a low hydrostatic pressure, because water enters by osmosis, water leaves by osmosis. That's what causes the mass flow, the mass transport of the sucrose solution from the source to the sink. Now, there are investigations that have been linked to translocation and working out the involvement of the phloem, which are tracers and ringing experiments. Tracers involves radioactively labeling carbon, and then that carbon is provided to the plant in the form of carbon dioxide. So by only providing the plant with radioactive carbon in the carbon dioxide, that means over time, all of the carbon-containing compounds in the plant should contain the radioactive carbon, because the carbon dioxide is taken in for photosynthesis and turned into sucrose, other sugars. So that means that you can take thin slices through the stem, and under the X-ray film, any radioactive carbon is going to show up. So in this way, you can track the route that the radioactive carbon takes through the plant, and that is how they could see where the phloem was, and that it must be the phloem that is transporting the sugars, because they are what lit up with that radioactivity under the X-ray. Ringing experiment is when a ring of bark, which will contain the phloem, are peeled and removed from a tree trunk. This can actually also be done on stems of smaller plants as well. And the result of removing the phloem is the tree starts to bulge here and here, so above where it's been removed. And that bulging, you can remove the liquid and analyse it to see it contains sucrose. But what this indicates is if you remove the phloem, the sucrose is made in the leaves further up, and when you take the phloem out, you get this bulging of liquid. The liquid contains sucrose, and that shows us that it must be the phloem transporting sugars, because when you remove them, the transport stops. Topic 4, and we begin with 3.4.1, DNA, genes and chromosomes. So comparing the DNA in a eukaryotic and prokaryotic cell is the first part of the spec, and this has been quite a common long answer question to do this comparison. So I've put it together here as a table so that you can see that direct comparison. And by the way, it's fine to present your answers as a table like this or a bullet point list in the exam, except for the essay. So eukaryotic DNA and prokaryotic DNA both are made up of DNA nucleotides. So that means they both have nucleotides that contain deoxyribose, the pentose sugar, phosphate group, and those nitrogenous organic bases. The nucleotides are joined together by phosphodiester bonds still, but the differences are eukaryotic DNA is longer than prokaryotic DNA. It's linear in shape, whereas it's circular for prokaryotic DNA, and then associated with the histones in eukaryotic DNA and not associated with histones in the prokaryotic DNA. So in eukaryotic cells, a DNA molecule is associated with the histones, forms a chromosome. So that's what we can see here. We've got the chromosome, and that is made up of tightly coiled DNA that is wrapped around these histone proteins. There is also DNA inside of the mitochondria and inside of chloroplast, which you learn about in topic 2. And the mitochondria within those organelles in eukaryotic cells contains DNA which is very similar in structure to the DNA that you get in prokaryotic cells. And the similarities are it's very short, it's circular, and it's not associated with proteins. So that is the properties of the DNA in mitochondria and chloroplasts, and that is a similarity to the DNA in prokaryotes. A gene is a base sequence of DNA, and it codes for the amino acid sequence of a polypeptide and functional RNA. And the mark scheme is really specific that you do say a base sequence of DNA, not just it's a section of DNA. And every gene occupies a fixed position, which means a certain place on a chromosome, and that's known as the locus. So for example, here we've got the locus for flower colour. The gene is for flower colour, that's the locus, meaning the location. You would always find it on the chromosome. A sequence of 3 DNA bases is a triplet, and that codes for a specific amino acid. And that is the fundamentals of the genetic code. There's 3 features of the genetic code that you need to know as well. So the genetic code is degenerate, it's universal, and it's non-overlapping. Degenerate then, this is based on the fact that there are 20 amino acids that the genetic code is able to code for. There are 4 DNA bases, guanine, cytosine, thymine and adenine. And just a note here, although I've abbreviated it, you are not allowed to abbreviate those in the exam. You have to write the full name. And therefore, 3 bases are needed to make enough combinations to code for at least 20 amino acids. And we can prove this mathematically. So 4 to the power of n, there's 4 bases. N is how many of those bases you would need to get enough to code for 20. So if it was 1 base, that would be 4 to the power of 1, which is 4. So that would mean we'd only be able to code for 4 amino acids. Guanine would code for 1, cytosine would code for 1, thymine would 1, and adenine, and that's not sufficient. If it was 2, that would be 4 to the power of 2, and that gives us 16 combinations. So that is not enough to code for 20 amino acids. 4 to the power of 3 is 64, so that is sufficient to code for 20 different amino acids, plus there are extra. And that is what leads to this property of the genetic code being degenerate. Because there's 64 combinations, which is more than the 20 amino acids, as a result, each amino acid is coded for by more than 1 triplet of bases. So that is what we mean by the genetic code being degenerate. And we can see that here in this genetic code wheel, where you would start on the inside, work your way out. So GUC, that codes for Valor. That's one of the amino acids. But GUU, GUA, GUC, all of those code for the same. By the way, this is a genetic wheel which always has the codons, which are mRNA. That's why we've got U for uracil instead of T for thymine. Now, it's an advantage that the genetic code is degenerate, because if a gene mutation occurs and it's a substitution mutation where one of the bases is now swapped and we have a different base, it might mean that the triplet of bases still codes for the same amino acid, and therefore, even though there was a mutation, you still code for the same amino acid, so it has no effect on the overall protein that is made. Universal is the next feature, and this means that the same triplet of bases codes for the same amino acid in all organisms. So that is your definition. Now, this has been used to our advantage because we can do genetic modifications, because we know that you can take a gene from one organism, put it into the DNA of another, and because the genetic code is universal, it'll still code for the same amino acids and make that protein. Non-overlapping means that each base in a gene is only part of one triplet of bases that codes for one amino acid. So therefore, each codon or triplet of bases is read as a discrete unit. So we can see here it's read as a discrete unit of 3, this is a discrete unit of 3, and so on. And that, again, is an advantage in case there is a gene mutation. Because if one base does get substituted and it does code for another amino acid, at least it's only affecting one triplet, whereas if it was an overlapping code, that one mutation would affect 3 amino acids. So 2 other key concepts are knowing what coding and non-coding DNA is within your DNA. So there are non-coding, multiple repeats of bases that you find between genes. But in addition to that, within a gene, you do also have sections that code and sections that don't. So within a gene, only some sequences, and these are the exons, code for amino acid sequences. And you also have sequences within a gene that do not code for sequences of amino acids, and those are known as the introns. So these non-coding sequences, you do have them between genes, but also within genes. And it's the ones that are within genes that are called introns, and those get removed during one of the stages of protein synthesis that we'll have a look at shortly. So 3.4.2 is DNA and protein synthesis. Now, I've mentioned this term codon, but the definition of codon is 3 bases on mRNA that code for a specific amino acid. The start codon is a particular set of 3 bases, and it's the 3 bases at the start of every gene that initiates translation, which is the second stage of protein synthesis. The stop codon is 3 bases at the end of every gene, and this causes a ribosome to detach in translation, and therefore it stops translation. So it stops protein synthesis. Genome and proteome. The genome is an organism's complete set of DNA in a cell, whereas the proteome is the full range of proteins that a cell is able to produce. So the genome should stay the same. It's not going to change unless there are mutations. Whereas your proteome, that does stay the same because it's the full range of proteins, but the reason it says is able to produce is not every cell is going to make all of the proteins that it's able to, but the proteome is the full range that the cell is able to make. Doesn't mean it does make all of those proteins. So next then, let's have a look at the 3 types of RNA. We have M, T and R. mRNA is short, it's single-stranded, and you find it in the cytoplasm and the nucleus. It's made during transcription, which we're going to see shortly. That's the first stage of protein synthesis. And it's a copy of one gene. So it's going to be a complementary copy to that DNA sequence in the gene. And as we said, 3 bases on mRNA are a codon, and that codes for 1 amino acid. tRNA, or transfer RNA, this is found in the cytoplasm, and specific amino acids become attached to transfer RNA molecules. Each tRNA is specific for 1 amino acid. Each tRNA molecule has an anticodon, which is these 3 bases here, which are complementary to the bases in a particular codon. You also have an amino acid binding site at the top, which is where a specific amino acid will bind, and that's how tRNA can bring a particular amino acid. This is involved in translation. It brings the amino acids to the mRNA on the ribosome. It's still single-stranded, it's folded to create what's known as a clover leaf shape. And those bases, as they're folded, will form hydrogen bonds, and that's what holds it in this structure. Now, I did say we talk about rRNA. That actually mainly comes up in topic 1. rRNA, you just need to know that combines with proteins to make ribosomes. So let's move on to protein synthesis. This happens at the ribosome, but it does actually, the first step where you make mRNA happens in the nucleus. So we're going to go through transcription and translation. Transcription is when a complementary mRNA copy of one gene of the DNA is created in the nucleus. As we said, the mRNA is much shorter than DNA, and that's because it's only a copy of one gene rather than the entire genome. And because it's shorter, that means it's able to fit through the nuclear pores in the nuclear envelope. It moves out, and then it can attach to a ribosome in the cytoplasm to be used in the next step. So if you were asked to describe transcription, here are your key marking points for transcription. First of all, the hydrogen bonds between the DNA bases break, and that causes the DNA double helix to unwind and the 2 strands to separate. Only one of the DNA strands acts as a template though. You will then get 3 mRNA nucleotides from within the nucleus, attracted to and aligning opposite their complementary bases. And in RNA, the complementary bases, just bear in mind, because it's RNA for mRNA, you have uracil instead of thymine. So any place on DNA where there'd be adenine, uracil would be the complementary base pair. Now that's not normally a mark, point 4, if it's a describe transcription question, but it's just worth noting. The enzyme RNA polymerase is then responsible for catalyzing the condensation reaction, which joins the adjacent RNA nucleotides together, and in doing that, phosphodiester bonds are formed. Once that mRNA is copied, it then gets modified, and then it's ready to leave the nucleus. So these modifications then, we actually call the mRNA before it's been modified pre-mRNA. Once it's been modified, then we call it mRNA. So the modification that happens is, once you've made your mRNA, and we've now got pre-mRNA, the introns get removed. And that's known as splicing. So the introns are spliced out, which just means they're cut out and removed. So then you're just left with the exons. And the exons are the sequences of bases on mRNA that code for sequences of amino acids. Now that happens in eukaryotic cells. It won't happen in prokaryotic cells because prokaryotes, such as bacteria, they don't have introns in their DNA, so they don't have to go through that step. The next stage is translation, and this is when the mRNA has now been modified, it leaves the nucleus, and it's going to attach to the ribosome, and tRNA is involved as well. So here are our key steps. Number one, I've just said. Number two, the ribosome, once that mRNA is attached to the ribosome, will start at the start codon and then it moves along the mRNA. But the key thing here is knowing that the mRNA attaches to the ribosome. Sometimes there's an extra mark for saying at the start codon, or it will move to the start codon. Mark number three, always going to be a mark. A molecule of tRNA has an anticodon complementary to the codon on mRNA, and that's due to complementary base pairing. Number four is always a mark. Each tRNA molecule carries a specific amino acid, and that's determined by the anticodon. The ribosomes will then join those amino acids, and a peptide bond forms between the amino acids. ATP is required for the formation of that peptide bond, but it's actually also required to join the amino acid to the tRNA molecule. The ribosome will then move along the mRNA to the next codon. The process repeats with new tRNA molecules bringing more amino acids and binding them. And this will continue until a stop codon is reached. So your absolute key marking points that you should always include would be number 1, number 3, 4, 5, 6, and 7. So 1, 2, 3, 4, 5, 6. This is often a 4 to 6 mark question. So they might be, here's your range, you have to say 4 of those, 6, or if it was a 6 marker, you'd have to say all of them. Next then we move on to 3.4.3, genetic diversity, and this is looking at mutations and meiosis. So genetic variation can be introduced in these 3 ways: through meiosis, mutations, and random fertilization of gametes. So meiosis is the type of cell division that creates genetically different cells, which are the gametes, so sperm and egg cells. And unlike mitosis, there are 2 rounds of division. In meiosis 1, that is when homologous chromosomes are separated. In meiosis 2, it's when the centromere splits and the sister chromatids are separated to the opposite poles. And by the end of both divisions, you have 4 cells. They are haploid, meaning only 1 copy of each chromosome, and they are genetically different. So I've just put this down here as a reminder of what haploid and diploid means. Haploid means 1 copy of each chromosome, and that is what you'd have in the gamete. So for example, the haploid number for a human is 23. Diploid means 2 copies of every chromosome. So for humans, the diploid number is 46. Now, the way that meiosis introduces genetic variation is the key focus here. You don't have to know PMATs, prophase, metaphase, anaphase, telophase, like you did for mitosis. You just need to know for meiosis how the genetic variation is introduced. And that occurs in 2 processes: independent segregation of the homologous chromosomes and crossing over between homologous chromosomes. You have to say homologous chromosomes for both of those. And if you're not sure what that means, homologous chromosomes are 2 chromosomes that have the same genes. So for example here, these 2 are homologous, they are the same size, same shape, and it's because they contain the same genes. You just have two copies of them, one that would have come from the mother, one from the father. Now, both of these processes here occur in meiosis 1, which is the first round of division. So let's go through independent segregation then. As we said, it happens in meiosis 1, and that is when the homologous pairs of chromosomes line up opposite each other at the centre of the cell, or the equator of the cell. But it's random which side of the equator the paternal and the maternal chromosomes will line up. They're always in their pairs, but it's random which side they go onto. And that matters because when the homologous pairs get separated, those chromosomes are pulled apart to the opposite end, depending which side of the equator they're on, will determine which chromosomes you get in these cells here, and then those get separated, the chromatids. So that is how the genetic variation is introduced. And there is actually a maths question linked to this quite often, using this formula here, 2 to the power of n, because you can use this to work out how many possible different ways are there for these homologous pairs to line up at the equator, and therefore how many different gametes could be made. And it's 2 to the power of n, 2 because we have pairs of chromosomes, n is for the number of homologous pairs you have. So for humans, that would be 2 to the power of 23, which is over 8 million. So because of independent segregation, there's over 8 million different ways that our homologous pairs of chromosomes can line up at the equator, and therefore over 8 million different potential gametes one human can make, just from independent segregation. But on top of that, crossing over can also occur. Now, crossing over is rare. That becomes important when you get to topic 7, learning about autosomal linkage. But for now, we're just going to go through what crossing over is and explain how it introduces genetic variation. So it also happens in meiosis 1. Again, it's linked to this homologous pair lining up. So we get the homologous pairs lining up at the equator. And when you have a pair next to each other at the equator like this, we call it a bivalent. So they line up and they form a bivalent. The non-sister chromatids, one chromatid is one of these arms of a chromosome. Non-sister means it's the chromatids from different chromosomes. So the blue and the pink here are non-sister chromatids. They cross over and entangle, and the exact point where they cross is called a chiasmata. Chiasma is the singular, chiasmata is the plural. And at each of those chiasma, you can get a break happening, and then you exchange that section of the non-sister chromatids. And as a result, when these chromatids separate to go on to make the final 4 gametes from one round of meiosis, you end up with new combinations of alleles in the chromatids because they've swapped sections of the chromatid. So if we just compare meiosis to mitosis, meiosis is 2 nuclear divisions versus 1. Meiosis, you get a haploid cell versus diploid. Meiosis introduces genetic variation. Mitosis, you get genetically identical cells. You could also be asked for meiosis to identify where meiosis is happening within an unfamiliar life cycle. And the key thing here is don't look for where gametes are made, because for some organisms, meiosis isn't making gametes. So instead, what you need to look for is where you go from having a diploid cell to a haploid cell. And they might write diploid or haploid, or they might write 2N, meaning diploid, or N. So that's what you need to look for on a diagram. And this is a bit blurry, but we can see here that we've got N, 2N, N, N, N. So where we go from the zygote to the zoospore, we've got 2N to N. That would be the meiosis stage because we've gone from diploid to haploid. Not here where we have that gamete being made. Now in this one, we can see everything on this side is 2N, everything on that side is N. So that means at this stage here, it is, I mean, it's labelled on, but if it wasn't, this is meiosis because we've gone from having a 2N cell to all of these being N. So that's what to look for. Diploid to haploid is always where the meiosis is occurring in a cycle. Gene mutations is another way that genetic variation is introduced. And a gene mutation is a mutation in a gene, but it means you're changing the base sequence of the DNA for that gene. And these mutations randomly occur during DNA replication. So there's a link here to DNA replication in topic 1, and there's a link here to the cell cycle in topic 2, because DNA replication happens in interphase during S phase. So these random mutations are more likely to occur if you are exposed to mutagenic agents, and they can interfere with DNA replication, and that's why. So mutagenic agents include high energy radiation such as UV light, ionizing radiation, which would be gamma rays and X-rays, and chemicals, so carcinogens such as mustard gas and cigarette smoke. There are 2 types of gene mutation that you need to know for topic 4. You do learn more in topic 8, but for paper 1 or for the AS, you only need to know deletion and substitution mutations, which is modelled here. So here is our original unmutated DNA sequence. A substitution is when one base is changed, and we can see that cytosine base in the original has been changed to adenine, and maybe that might still code for the same amino acid because the genetic code is degenerate, or maybe not. A base mutation that is a deletion is when one entire base is deleted, and we can see that one of the cytosines has been deleted here, and all of the bases after the point of that mutation have now shifted left. And this is known as a frame shift because all of the codons after the point of mutation have now changed. So that means we're going to code for all of those, probably different amino acids. So a deletion mutation is far more harmful, potentially, than a substitution mutation because more amino acids will be coded for incorrectly. You can also get chromosome mutations, and this is mutations in the number of chromosomes, and that can happen during meiosis. Now, a chromosome mutation, if you're asked in the exam to name the chromosome mutation, it's non-disjunction. That's come up quite a lot recently, and sometimes students aren't sure what they're meant to say the name of the chromosome mutation is, and the name of it is non-disjunction. So non-disjunction, what this means is during meiosis, either the homologous chromosomes in meiosis 1 don't separate equally, or if it's meiosis 2, the sister chromatids don't separate equally. So as a result, you end up with more either chromosomes or chromatids going to one cell, and the other cell has a lack of chromosomes. So that is the non-disjunction chromosome mutation. Now this is just going to show you it, so you can visibly see the outcome. So in this scenario, it's in meiosis 1. The homologous pairs have lined up at the equator, and for some reason the spindle fibres are all coming from one of the centrioles, and that means all of those homologous pairs are going to be pulled to this cell. This one has got none, so that cell isn't going to survive, and this one has now got an extra copy of every chromosome in it. Meiosis 2 is when the sister chromatids separate, but because we had 2 copies of every chromosome, we end up with 2 copies in our gametes. So we have diploid gametes here instead of haploid. So this is one example of non-disjunction where we now have an extra copy of every chromosome in that gamete. Now, if that gamete was to fuse with a haploid gamete where there are no mutations, then 2 plus 1 equals 3. So we now have a triploid cell. So this is one example of non-disjunction. And this does happen quite commonly in plants, and plants can still grow and survive that, but this isn't seen in humans. Triploid would be fatal. That wouldn't lead to a fetus and a baby that could actually be born. Now, another type of non-disjunction we can see here, it's still happening due to the centrioles only producing spindle fibres on one side, but meiosis 1 happened normally. This time it's meiosis 2, where the sister chromatids are both pulled to the same side. This one, there was no chromosome mutation, no non-disjunction, but in meiosis 2, This one there was, all of the sister chromatids moved to one side. So again, we end up with that diploid gamete and one gamete with no chromosomes. Now, another type of non-disjunction that can happen is if only one of the chromosomes, the spindle fibres, are attaching at one side. And as a result, both copies of only that chromosome move to the same cell. So we still end up with a haploid gamete, but you have one extra chromosome. So it's haploid, we've got one copy of all the chromosomes, except for this one, we've got one extra. And this one's haploid, one copy of all of the chromosomes, except we're missing one. So it'd be described as n+1, and these are n-1. And if you then had a gamete like this, combining with a haploid gamete, you end up with a diploid zygote plus one extra chromosome. And that's actually what happens in Down syndrome. There are 3 copies of chromosome 21 because of this type of non-disjunction chromosome mutation. If it was the n-1 gamete, then you'd end up with a zygote that was still diploid minus 1 chromosome. Next then we move on to 3.4.4, genetic diversity and adaptation. And genetic diversity is the number of different alleles of genes in a population. And this is one of the key driving factors of natural selection, because you have to have genetic diversity in order to select which allele is going to be passed on. So a gene pool can be described as all of the genes and all of the alleles in a population at a particular time. The allele frequency is the proportion of organisms within a population carrying a particular allele. We're going to see these key terms come up as we go through natural selection and evolution. So natural selection is the process that leads to evolution. Your definition of evolution is the change in allele frequency over many generations in a population. And this helps to ensure that you have a population of species better adapted to their environment. And these adaptations could be anatomical, physiological, or even behavioural adaptations. So natural selection is random mutations occur. That's always your first marking point. If it's an applied example, you have to state which allele has been made. So in this case, it might have been there was a random mutation that created the white mouse or the black mouse, whichever one it was. That introduces genetic variation. Those are often a combined mark. Some mutations might be harmful, but some are actually advantageous. So if an individual did have that allele, they might be more likely to survive. And if they're surviving, they're more likely to reproduce. And if they reproduce, then they're passing on that new mutated allele to their offspring. And if this repeats over many generations, most of the population or a lot of the population will now have this allele in their DNA. So that means the frequency of that allele has changed over many generations, which was our definition of evolution. Now, there's 2 different outcomes that you learn about, which is types of selection or outcomes. You do learn a 3rd type in topic 7, but there's only 2 that you need to know for paper 1 or for your AS. And one of those is directional selection. And this is when the allele that is providing the selective advantage is one of the extreme traits. So it could be very, very dark fur if it was the mice, or very, very light fur if it was the mice. Or the example that you actually need to know is antibiotic resistance. So bacteria that are very, very, very resistant compared to not very resistant or not resistant at all. The very resistant ones are surviving. Their alleles are then being passed on. And the outcome is we see this shift from the original red line showing us the original distribution of the different alleles in the population. Now we can see there's been a change in the allele frequency because most individuals now have that extreme trait. Stabilizing selection is if there's no change in the environment. So whatever was the most common or the modal trait, that is still the allele that's providing the selective advantage, and it still therefore remains the most common. It's just over many generations of natural selection happening, there's still a change in the allele frequency because now even more individuals have that modal trait and the extreme traits have been lost. So in terms of standard deviation, the spread of data around your mean, we've lost those extremes, so the standard deviation has decreased. The example linked to this is human birth weight. So a middling birth weight is a selective advantage, because if you're much, much lighter, then potentially you were premature, and that means maybe you're less likely to survive. And if it's a very, very big baby, you're more likely to have complications with the birth. So the middling birth weight is a selective advantage. Next then we've got 3.4.5, species and taxonomy. The definition of a species is two organisms that are able to breed and make fertile offspring. But the key thing is, can they breed and make fertile offspring? If they can, they're part of the same species. And species must reproduce and pass on advantageous alleles in order for the entire species to survive. And courtship behaviour, we did say behaviour is an adaptation. Courtship behaviour is genetically coded for, it is an adaptation, and it is essential for successful mating and species recognition to help the entire species survive. So what courtship behaviour means is, it's the different behaviours that animals demonstrate as a way to attract a mate. And each species has their own unique sequence of behaviours to ensure successful mating. And what we mean by successful mating is if the two individuals are mating and produce offspring. Now, normally the courtship ritual is performed by the male, and then the female selects which male they want to reproduce with. And the behaviours are often a sequence of behaviours. And it's not necessarily just behaviours, it could be sounds, the release of pheromones, which are chemicals. So it could be dance moves, displays of colour and feathers. There's lots of really extravagant courtship behaviours in plants. And in fact, if you just search birds of paradise courtship behaviours, there's some really cool David Attenborough videos on YouTube for that. So females will observe the courtship ritual and decide if they want to mate with that male. So the reason this is important is, as we said, it's to ensure successful reproduction, which means mating and producing offspring so that the whole species can survive. And the reason it increases the likelihood of an offspring being produced is it enables you, first of all, to recognise members of the same species. Now that sounds fairly obvious and not needed if you're thinking about humans, but for other animals, they do rely on this to make sure they are the member of the same species before they reproduce. Secondly, in order to be able to make offspring, they'd need to be mating with a member of the opposite sex. So that's the first two things that courtship helps animals to do. Recognise members of the same species, recognise the opposite sex. Next is synchronising mating behaviour. Now what this means is animals will do the courtship ritual and they'll be able to recognise what that ritual means only if they are producing gametes. So males will only do the dance when they are producing gametes. Females will only recognise and respond to the courtship ritual when they are releasing eggs. And again, that then means if they reproduce, you're more likely to have offspring because both the male and female are producing gametes, which could then fuse together to make an offspring. Now, on top of that, this isn't to ensure successful mating, because this is now saying for some animals, there's an extra importance to courtship behaviour, and that's to ensure the survival of their offspring. And that's because these courtship rituals can help to form a pair bond, which means the two parents should stay together. And we see this in penguins because it's a massive advantage to the survival of the offspring if they have a mother and a father, because one of them stays to look after the young while the other goes to find food. It also helps to enable the female to select a healthy male to mate with, and therefore their offspring is more likely to be healthy and therefore increases the survival of their offspring. And the logic behind this is, if a male is fit and healthy, then they'll have more energy that they can put into their dance routine, or their feathers will look brighter in colour, they might look better conditioned. So that is the idea behind that one. Next then is the binomial system. Bi meaning two, nomial meaning name, so the two name system. And this is when you use the genus and the species to name the organism. And this is the universally used, meaning it's the same across the world, the universal scientific way to identify an organism is the binomial system, genus and species. And this is useful because, first of all, you can share data, but also it gives you more information than the common name would. So, for example, we've got two birds here. The common name is New Zealand robin, and then this is a robin. And you might think, oh, they're both called robins, so they must be closely related. But if we have a look at their name using the binomial system, their genus and species is different. So they're not actually even closely related. So it's useful to be able to see, are different species part of the same genus? Because if they are, that means they're closely related. If they're not, then they're not closely related. And that links us into phylogenetic classification. And this is arranging species into groups according to their evolutionary origins and relationships. So it's a way for us to see, for different species, how closely related they are. Do they share recent common ancestors? And that's what these phylogenetic trees show. So humans, for example, in this tree, we're most closely related to chimpanzees because we share the most recent common ancestor. Then as we go back in time, so we're going back in time this way, after that, here is the next most recent common ancestor. So that means after chimpanzees, we're most closely related to these two. And then we can go back again and see it's then these, and so on and so on. So every fork or branching point represents a common ancestor, which means that is the species, anything that branches off, evolved from. Now, linking to classification still, individuals can be classified according to their physical and biochemical similarities, and classification systems are an example of a hierarchy. And that is a really common 2-mark question. What is meant by hierarchy? And it's when you have smaller groups arranged with larger groups. That's mark 1. Mark 2 is there's no overlap between the groups. So what that means is we've got an example of a hierarchy here, which you're probably familiar with from GCSE, and you do still need to know this for A-level. We can see that within a genus, there are lots of smaller groups. And that's because in one genus, there'll be lots of different species, but there's no overlap between those species. They're all different species, they just all belong to the same genus. And then we can go up again and say the same thing with genus. Within a family, there's lots of different genuses, but there's no overlap between them. 3.4.6 is biodiversity within a community. And biodiversity refers to the variety of life, and it can be measured on different scales. It can relate to a small local habitat, such as a pond or a woodland, or the entire earth. And there's different terms to classify or to describe biodiversity. We've got species richness. That definition is the number of different species present in a community. So it just tells you how many different species there are. Index of diversity gives you more information than that because it's looking at the relationship between the number of species, so the species richness, but it also takes into account the number of individuals within each species, which is far more helpful because then we can get an idea of, are any of those species at risk of going extinct, because we now know the number of individuals within each species. And biodiversities can be used to describe, as we said, local habitats or the entire earth. And a low biodiversity isn't always a cause for concern because it might be naturally there is just a very, very low biodiversity because there's such harsh abiotic factors. So, for example, in the Arctic or anywhere with extreme abiotic factors. The concern is when you have an area that had a high biodiversity and it's starting to decrease because something is causing individuals and maybe entire species to die out. And often that is due to human activity. Now we can calculate this index of diversity using this formula. They give you this formula, but they don't always tell you what the capital N and lowercase n is. Often they do, but I'd still say learn it because the most common mistake students make on this is misremembering what the lowercase n actually means. So capital N is the total number of every organism alive in that area, whereas lowercase n is just a population size of one species. The best way, though, to get an idea of this is we'll go through an example. Now with this formula, typically you will get a value that is between 1 and 10. It doesn't have to be 10 as the maximum, but usually you get an answer somewhere between 1 and 10. So if you do this in the exam using this formula and you get an answer like in the 50s or 100s, you've gone very wrong somewhere. Recalculate it, try again. So to model this then, capital N we said is the total number of organisms. So that is the sum of that column. We've got 4 species, added all the individuals together. Lowercase n is the population size of each species. And we have to do n times n minus 1, and then it's the sum of that for every species. So n is 6, so we need to do 6 times 6 minus 1, which is 5. 6 times 5 is 30. Then for this one, 3 times 3 minus 1, so 3 times 2 is 6. 12 times 11, 132. 4 times 3 is 12. And then the denominator is the sum of all of those. So you do have to work this out for every single species they give you in the table. Add that all together and that gives us 180. Then the numerator is the total number of individuals, which in this case is 25. So we do 25 times 25 minus 1, which is 25 times 4. And then that comes to 3.3 when we finish the rest of that equation. Farming is one of the examples of human activity that can reduce biodiversity. And within the specification, it says you need to be awareness of the balance of we need farming to provide food for humans, but there needs to be a balance between having that farming and not having too negative an impact on the biodiversity. So some of the ways that modern farming techniques might reduce the biodiversity are the removal of habitats. So that could be removing hedges and putting in fences instead, or just not having a hedge there at all. It could be removing habitats by cutting down a forest and then just planting crops. It could be using a monoculture, which means just growing one species. And if you've removed a whole range of plant species and replaced them with only one, reduce the species richness, but also the variety of food available for herbivores. If pesticides and herbicides are used, that is going to kill animals and plants, which again is reducing the species richness. And the overuse of fertilizers can cause harm in aquatic ecosystems as well. But 2 key marking points that always come up linked to this concept of farming techniques and how this can reduce biodiversity is there will be fewer habitats. So fewer habitats and fewer food sources. Not quantity of food, it's food sources, meaning one plant might eat grass, another plant might eat daisies. I'm just doing 2 random examples. So if you only have grass now, any of the animals that ate daisies can't survive there. So it's fewer habitats, fewer food sources if you have fewer plant species present. Next then, investigating diversity. So genetic diversity refers to the variety of alleles within or between species. And you can assess this by comparing the frequency of measurable or observable characteristics. So for example, the flower colour. But this can be less reliable because characteristics are also influenced by the environment as well as the genetics. So more accurate ways to compare are comparing the DNA base sequence. This is a direct measure of how similar or how different your DNA is. You could also do base sequence of mRNA because that is copied from the DNA. Or it could even be the amino acid sequence in proteins because amino acids are coded for by DNA. However, we do need to bear in mind for this one, because the genetic code is degenerate, 2 organisms might have the same amino acid sequence for a particular protein, but their DNA base sequence might be different because the genetic code is degenerate. So the most accurate measures are comparing DNA base sequences or mRNA base sequences. So when measuring variation within a population, this is where we get the concept of sampling, because we'd take samples because it'd be too time consuming to literally measure it for every individual organism, and it might not be very accurate. So to investigate variation scientifically by doing sampling, you have to make sure that you're doing random sampling to avoid bias. You must take large samples to ensure the results are representative, and then calculate a mean to summarise the data. You can then also go on to do standard deviation and even statistics. That is going to be the most useful, because if you do a statistic, you can see if you have a significant correlation or a significant difference, depending on what sort of pattern it was you were looking for. So that takes us to the end of topic 4. Hopefully you found it helpful. If you do want even more help with mark scheme specific key points, or if you want to have examiner tips, essay links, recall questions, then go for my A-level notes, flashcards. That is all about mark scheme key points, learning those terms. So I'll link those below, and you can click the QR code or scan the QR code at the top. That's it though for today's video. Hope you found it helpful, and I'll see you in a video very soon.